Okay, welcome everybody. This is Chapter 2. What we're going to be reviewing here is the subject of chemistry, and we have to understand a little bit about chemistry in order to study the human body because the human body, along with virtually everything else in the universe, is made up of either matter or energy, and chemistry addresses the interaction between matter and matter and energy. So, first off, let's define what matter is. Matter is anything that has mass and occupies space. Um, and the other entity that fills up the universe is energy. And one of the things that we need to understand is that when the universe was created in an event that scientists believe is called the Big Bang, that all the stuff in the universe, all the physical stuff in the universe was created in one um, gigantic event, an explosion that produced all the energy and matter in the entire universe. And energy and matter are related to each other. Energy and matter can be interconverted, but we can't make energy or matter disappear, and we can't make them appear out of nothing. And that's one of the laws of thermodynamics. The first law, in fact, states that energy and matter can't be created or destroyed, but only converted into each other. The second law of thermodynamics states that every time there is an energy change, such as from um, kinetic energy, the energy of movement, to potential energy, the energy of position, electrical energy, what have you. Any type of energy transformation will always produce a little bit of energy lost as heat, and this heat will go towards randomizing the universe. And so the second law of thermodynamics is sometimes stated that the universe tends towards maximum entropy or disorder. Or if you want to think about it in a different way, the the universe as a whole is losing useful energy, and as a result, if you look at any part of the universe, if you don't put any energy into it, it'll try to arrange itself in a way that reduces the amount of energy needed to maintain that particular system. So things tend to take the easy way out. Examples of that include a smoke ring filling a room, a drop of food coloring diffusing across a glass of water, um, water running downhill. All these are examples of a system losing useful energy and arranging itself in a low energy state. Weight is the pull of gravity on mass. Mass does not have weight without the force of gravity. And there are three states of matter we need to become familiar with. Solid, where the matter has a definite shape and volume, and the atoms that make up the solid don't move very much. They basically vibrate around fixed points. Liquids, which have a changeable shape and a definite volume, and this is due to their kinetic energy, which allows the molecules in the liquid to slide past each other, and as a result form to the shape of their container. And then gas, which has a changeable shape and volume. Gas has even more kinetic energy than liquid, and as a result, the molecules in a gas can not only move past each other, but they can occupy potentially um, every square inch or every cubic inch of the container in which they're found. And as matter transitions between these three states, what happens is that we gain kinetic energy. So the solid has the least kinetic energy, the liquid has an average amount, and the gas has the most kinetic energy, energy of movement. Now, energy is the capacity to do work or to put matter into motion, and there are different types of energy. Kinetic energy, which is the energy of movement, and potential energy, which is the energy that results as a function of position. Energy can be transferred between these two types, and when that happens, what we lose is a little bit of heat as a result. As an example, okay, you can look at this guy that is about to pull this bow back. So let's get a pin here. Now, what's going to happen is when he pulls this bow back, chemical potential energy, which is present in his muscles, is going to be converted into kinetic energy that will cause the muscles to tighten. And when they tighten, that movement will pull his arm back, which will pull the bowstring back. Okay, and now in that tightened bow, let's draw a tightened bow here. The dotted line would be the tightened bow. What we have now is potential energy that's been transferred from the chemical potential energy in his muscles to the kinetic energy of his muscles to the potential energy of the bow. Now what will happen is that the arrow will be pulled back, okay, and then he will release that bow. The stored potential energy will be transferred into kinetic energy as the bow snaps forward. Let's draw the snapping forward in red. And what will result is that the arrow will have that kinetic energy transferred to it, and it'll head towards the target. When it hits the target, it'll make a noise. That's a form of energy. And it'll produce some heat, which is another form of energy. And the rest of that energy will be distributed through the target. The target may shake a bit. Okay? And that's an example of an energy transformation. And at every step of that energy transformation, a little bit of energy was lost as heat, and that went out into the universe and helped to randomize the remaining components, the remaining matter and energy. The equation that relates matter to energy was developed by a gentleman named Einstein, and it looks like this: E equals the equal sign there, m for matter, c squared, where c is the speed of light times itself. Okay, so you can see it doesn't take much matter to produce a lot of energy, because the speed of light is a really, really huge number, and if you square it, it's an even bigger number. And this is the equation that led ultimately to the development of nuclear power and the atomic and hydrogen bomb. So let's look at some different forms of energy. Chemical energy is stored in the bonds of chemical substances, where I termed it chemical potential energy. Electrical energy results from movement of charged particles. Essentially, one of the things that happens when a charged particle moves from one place to another is that it generates both an electrical and a magnetic field, and those two fields will always be found together. Where there's a moving electrical field, there's going to be a magnetic field, and where there's a moving magnetic field, there's going to be an electrical field. Okay, so those are types of electromagnetic radiation, which is another form of energy, electrical energy, that can be used to do all kinds of things. Then there's mechanical energy, which is directly involved in moving matter. And then radiant or electromagnetic energy, which travels in waves. Examples include visible light, ultraviolet light, X-rays, radio waves, infrared. And then what we find is as we move through the electromagnetic spectrum, let's draw this in black, we find the visible light here, Roy G. Biv, right? Red, orange, yellow, blue, green, indigo, and violet, somewhere in the middle of the spectrum. And then as we move to the right, the wavelength gets smaller. We find UV radiation over here. Okay. We find X-rays. And then if we get all the way over to the very far end of the spectrum, we actually sort of transition from a wave into a particle. And so we find things like electrons, which we'll indicate with an E with a negative sign next to it for reasons that'll come clear later. Protons. And neutrons. These are what are called subatomic particles. And then on the other end, as the wavelength gets bigger, we find infrared or IR. And then we find radio waves. Radio. and then the energy continues to shrink from there. Now, notice that I said that the energy gets the energy content of the electromagnetic radiation increases as we go to the right and it decreases as we go to the left. And you might ask, well, why is that? Well, that's because of two other equations that that are given to us from physics. One is that frequency is equal to 1 over wavelength, where that funny looking symbol there, lambda, stands for wavelength, and that little funny looking V over here stands for frequency. And then there's another equation which comes to us from physics that states that energy is equal to a constant, which is called Planck's constant, times frequency. So the higher the frequency, the higher the energy, and the smaller the wavelength, the higher the energy. Kind of interesting. And y'all know this. I mean, if you've been exposed to intense amounts of UV radiation, it'll burn your skin, whereas if you're exposed to radio waves or infrared, that's going to have negligible damaging impact on biological tissue. And then, of course, visible light, which we're all familiar with, is relatively low in energy as well. So that's the electromagnetic spectrum. And again, what electromagnetic means is that everywhere that you see an electrical field, you're going to see a magnetic field moving with it. Energy can be converted from one form to another, but this conversion is inefficient. Some energy is going to be lost as heat, and that energy is essentially only useful in randomizing the universe. And this is what the second law of thermodynamics is trying to say, which is that the universe is tending towards maximum entropy because of this heat that's coming off from all the energy changes in the entire universe. An example of some energy conversions that you may or may not be familiar with. If you've ever seen pictures of Las Vegas, okay, I can draw you an example of how Las Vegas gets its electricity. So let's get a blue pen here. So there's a lake called Lake Mead that was created a little bit north of Las Vegas as the result of a dam that was built, Hoover Dam. The water that builds up behind the dam, as you can see here, as it gets higher and higher above the surface of the earth, the potential energy increases, right? You all know that if you're standing up way high on a building, you've got more potential energy that can be turned into kinetic energy when you jump off the building than if you're down, say, on the first floor. And the same is true with the water, right? The water's building up behind the dam, and the potential energy is building. And so the way Las Vegas gets its juice is that inside the dam are little turbines, and these turbines have on them magnets, and we'll indicate these magnets in green, let's say. Okay. And what happens when the water flows from a high place to a low place over the dam is that these magnets, they spin around a copper coil. And the copper coil is basically in the center of the turbine, so let's draw that. So there's the center of the turbine right there. There's the copper coil. Okay. So as this spinning magnetic field, which is produced by the water, is taking place, as the water flows over the dam and down into the river below, this wheel will spin, and as this wheel spins, that moving magnetic field will produce an electrical current in the copper wire, and as a result, what will happen is that electricity is produced, which is moving negative charges, moving electrons, and that's what powers Las Vegas, Nevada. That's where it gets most of its electricity, through hydroelectric power. A very clean form of energy. Okay, so next time you go out to Las Vegas and you see all those lights, that's where it comes from. Hydroelectric power. That's another beautiful example of energy conversion, and also a demonstration of how a moving magnetic field can produce an electrical current. Kind of interesting. Okay, elements are essentially the matter of the universe. Okay, elements are made up of atoms, They can be broken into simpler substances they cannot be broken into simpler substances by ordinary chemical means. Each is going to have unique properties that are detectable with our senses or are measurable with instrumentation, and they'll all have unique chemical properties, and these chemical properties dictate how these atoms interact with each other, basically what kind of chemical compounds they can form. Um, all the known elements are shown in a format known as the periodic table, which was developed by a gentleman named Mendeleev. And what he was able to deduce as a result of understanding atomic structure is the mass of each of these elements and their chemical behavior. And he was able to arrange those in a very easy to understand format. So let's talk a little bit about the building blocks of elements, which are atoms. Atoms are the smallest unit of an element that still have the chemical properties of that element. They're unique for that element, and they give the element its physical and chemical properties. And the concept of the atom is actually goes all the way back to the ancient Greeks, where a gentleman named Democritus was looking at a rock one day, and he hypothesized that he could take that rock and break it in half, and both halves would still look and behave like a rock, and he could take those halves of rocks and break them in half, and the properties would still be there. But he figured that he would eventually get to a point where he couldn't make a smaller unit of matter that would still retain the properties of that original rock. And he termed that smallest particle the atom. And that's the working definition of the atom today, the smallest particle of an element that contains the chemical properties of that element. Atomic symbols are used to represent the elements, and these are one or two letter chemical shorthands for each element. And so here we can see the four elements that make up about 96% of the body mass. That's carbon, hydrogen, oxygen, and nitrogen, chone. A nice way to remember that. Those are the atomic symbols for each of those elements. Okay. 9 elements make up about 4% of the body's mass, and those include calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, iodine, and iron. But just because these are present in small amounts in the human body doesn't mean they're unimportant. For instance, if I were able to snap my fingers and remove all the iron from your body, you would die because you wouldn't be able to make usable chemical energy to keep you alive, and your blood wouldn't be able to carry oxygen, which your cells need in order to produce that energy. So just because an element is present in tiny amounts doesn't mean that it's unimportant. Okay. There are some trace elements that are present in tiny amounts. They make up about 0.01% of the body's mass, and most of these act in concert with enzymes, which are proteins that in living systems help chemical reactions take place. Some of these include chromium, copper, fluorine, manganese, silicon, and zinc. So let's take a look at our understanding of what atomic structure is. And we have to preface this by saying that in science, one of the things that we like to do when we have a body of information regarding a particular phenomenon is create what we call a model of that phenomenon. And the model of the atom reflects only what we know about the atom right now. And it's got some holes in it. There's some things about this model that we don't completely understand, although we're working towards a bigger understanding of the atom all the time. But there's two competing models for the atom, the orbital model and the cloud model. And essentially what the orbital model states is that the bulk of the mass of the atom is contained in the nucleus, which is made up of protons and neutrons, each of which weigh one atomic mass unit, and they contain, again, most of the mass of that particular atom. The electrons are much, much smaller, but they move around the nucleus at the speed of light, and they have a negative charge. The protons have a positive charge, and the neutrons have no electrical charge. So this is one model of the atom. Another model of the atom indicates that the electrons don't move around the nucleus like planets around the sun, but rather they occupy a physical space around the nucleus that'll have any of a variety of different shapes. Many of the orbitals have a spherical shape, but some of them have more unusual shapes. Some are shaped like dumbbells, some are shaped like figure eights, some are shaped like snowflakes. And we think that it's this cloud model that probably comes closer to explaining how atoms actually behave. Now, this is an example of an atom called helium. Helium has 2 protons and 2 neutrons in the nucleus and 2 electrons orbiting the nucleus. As a result, the electrical charge on that atom is 0. And the reason for that is that the number of positive charges is equal to the number of negative charges. And you'll find that in any element, that's the case, that the element itself has no electrical charge. Now, sometimes what can happen is that an atom can gain or lose electrons and form what are called ions. A cation is a positively charged atom, one that has lost one or more electrons, and an anion is an atom that has gained one or more electrons. Now, you might ask, well, where do these electrons come from and where do they go? They go to other atoms. Or they participate in what we call chemical bonds in the case of atoms that are attached to each other covalently, but we'll get into that in a minute. So you can have ions, you can have atoms, but elements in their pure form have no electrical charge. And so how do we indicate the number of protons in the nucleus of an element and the number of electrons orbiting that nucleus? We do that with the atomic number. Okay, so the atomic number of helium is 2, and that tells us that there are 2 protons in the nucleus and 2 electrons orbiting. The atomic mass is the total of the protons and the neutrons in the nucleus. So the atomic mass of helium is close to 4. Now, the reason that I say it's close to 4 is because when all the matter in the universe was created at the Big Bang, not all the matter was created quite exactly the same. Some of the matter for each element has some extra neutrons in the nucleus, and they form what are called isotopes. Now, sometimes these isotopes are unstable in that they will over time decay into other elements, and sometimes these isotopes are stable and they are always going to behave chemically just like the normal element. It's just that they're going to have a little bit of extra mass. The reason that they're important in living systems is because we can use isotopes that give off radioactive energy as a way to essentially examine the metabolism of living tissues. Okay, different elements are going to contain different numbers of subatomic particles. Hydrogen is the simplest element. It has a single proton, no neutrons, and a single electron. Well, lithium has 3 protons, 4 neutrons, and 3 electrons. So the atomic number for hydrogen would be 1, as would the atomic mass. It would be a little bit bigger than 1 because there are some hydrogen atoms that have neutrons in them. Those are isotopes. And lithium, right, with 3 protons and 4 neutrons is going to have an atomic mass of close to 7, but an atomic number of 3. Okay, that's indicating the number of protons in the nucleus and the number of electrons orbiting it. Okay, now I told you that one of the ways that we understand how the different elements behave is that we have a device called the periodic table, and the periodic table is shown here, and what it tells us is the chemical symbol for the element, and it also tells us the number of valence electrons. Those are the electrons that are in the outer shell of that particular atom. And the reason that that's an important thing to know is because that's what dictates the chemical behavior of each element. In addition to that, if we look at the periodic table, we can also see the atomic number and the atomic mass. And some periodic tables are even nice enough to show us how the orbitals fill, the order in which we load the electrons into each of these atoms. And this periodic table gives us a lot of that different advice. So if we were to look, say, at this first column of elements down here, see this 1A at the top indicates that there's 1 electron in the outer shell of each one of these elements. 2A indicates that there's 2 electrons in the outer shell of each one of these elements. If we jump over here, there's 3 electrons in the outer shell of these, 4 here, 5 here, 6 here, 7 here. And then in this column, what we have are full outer shells, meaning that these elements are not going to chemically interact with other elements because they don't need to gain or lose or share electrons in order to form compounds. They're stable, they're low energy, just as they are. The first member of this column of the noble gases, which is what this column of elements is called, is helium. Helium has 2 electrons in its outer shell, and as a result, that outer shell is full. And essentially, the subsequent shells for the atoms that we're interested in this course have a maximum occupancy of 8 electrons. So neon has 8 in its outer shell, as does argon, krypton, xenon, radon, and so forth. Okay. And what that means for each of those elements is that they don't chemically react with anything. Now, how about these elements that don't have full outer shells? What's their story? Well, if we look at the group 1A elements, okay, these are called the alkali metals, okay, and what they like to do is to dump this outer electron because that gives them a full shell on the outside because the shell underneath the valence shell is completely occupied. So in the case of hydrogen, hydrogen is kind of an exception. Hydrogen can either gain or lose an electron in order to form chemical compounds, or in some cases it can share because it only has the first shell available. Lithium, however, has a full first shell, okay, so there's 2 electrons in its first shell, and then in its second shell it's got 1 electron. Down here with sodium, the first shell contains 2 electrons, the second 8, and the last shell 1. That's why we have the atomic number of 11. Potassium, first shell 2, second shell 8, third shell 8. 8 and 8 is 16, plus 2 is 18, and then the outer shell has 1. And so as we go down the column, we keep gaining a shell, a full shell, but remember that the outer shell is always going to have 1 electron. So what the members of this column of elements like to do is surrender their electrons in order to form mostly ionic compounds. And so this is why a lot of these elements are found in salts, okay, which are ionic compounds, which are formed when one element gives up its electrons and another element accepts them. Okay, the alkali earths are the next column over. They each have 2 electrons in their outer shell: beryllium, magnesium, calcium, strontium, barium, radon, and so on. And then over here, and again, what do these elements like to do? They like to give up their outer 2 electrons in order to form salts for the large part. These are called the alkali earths. And if we jump over to this side of the periodic table, what we find are elements in this column that have 3 electrons in their outer shell. Okay, boron, aluminum, gallium, indium, tellurium, and so on. And these elements can form ionic compounds sometimes. Sometimes they can form covalent compounds. Over here, the group 4 elements have 4 electrons in their outer shell. They largely form covalent compounds. Basically, you can see where the green and the black kind of form a stair step right here. Everything that's on the stair step is what we call a metalloid, meaning that it can form compounds similar to the kind of compounds metals form. Metals are everything to the left of this stair step, and nonmetals are everything to the right of this stair step. So everything over here that I'm tracing, these all are nonmetals. Okay, metals tend to give up electrons. Nonmetals tend to pick up or share electrons. When these compounds form chemical when these elements form chemical compounds with each other, they tend to share electrons. When they form compounds with Elements that are over on the other side of the table, left side of the table, it tends to be a giving or taking of electrons. So we tend to formally call ionic bonds. Now, these guys in the middle, these are called the transition metals, and they tend to give up electrons. Their chemistry is much more complex than the stuff that we're interested in. If we wanted to sort of circle the elements that we're going to be dealing with mostly in this course, we kind of draw a box around what we're interested in here. These guys, and over here we're going to be dealing with hydrogen a bit, some with sodium and potassium, some calcium and magnesium, okay. So the guys that are covered by the red boxes, and then a little bit of iron, the guys that are surrounded by the red boxes are the ones that we're going to worry about mostly in biochemistry. Okay, you're not going to run into many of the other elements much, so you don't have to worry about their chemistry that much. But I did want to show you what the periodic table looks like. Okay, so that's essentially what we're going to be working with. The atomic number, the mass number, and the atomic weight are the critical identifiers for each element. They give a picture of that element. They allow identification. The atomic number, remember, is the number of protons in the nucleus, and it's written as a subscript to the left of the atomic symbol. As an example here, lithium with its atomic number 3. The mass number is the total number of protons and neutrons in the nucleus. It represents the total mass of the atom, and it's written as a superscript to the left of the atomic symbol as shown here. Okay, so if we were to draw the complete picture of lithium, we could do it like this. Let's do that in red. Lithium would be written like so, Li, with the atomic number down here, 3, and the atomic mass up here, 7. Okay, and that's the complete picture of that element. Isotopes are structural variations in atoms that differ in the number of neutrons in the nucleus. The atomic numbers are the same, but the mass numbers are different. However, the chemical behavior is the same because the number of valence electrons isn't any different. Average of mass numbers of all the isotopes of an atom gives us what we call the atomic weight. And so one of the things that we should do before we head on to a further discussion of radioisotopes is we should real quick look back at the periodic table just for a minute, and I wanted to point out something with regard to carbon. Now I know this is a little small to see, but if you refer to the periodic table in your textbook, one of the things that you'll note when we look at carbon is that it has an atomic mass of a little bit greater than 12, 12.01 to be exact. So let's kind of call that out down here. So there's carbon, okay, at 12.01. And of course the atomic number is 6. Now, why is it 12.01? Why isn't it exactly 12? And the reason for that is that if we were to go out and collect a large number of carbon atoms and weigh them, we would get a weight that's a little bit greater than 12 atomic mass units per atom. Or, if we want to deal with a measurement we can understand, if we were to collect an extremely large number of atoms, in fact, let's say that we were to collect 6.02 times 10 to the 23rd carbon atoms, which is a lot, and we were to weigh that on a balance, they would weigh exactly 12.01 grams. Okay, so that's one of the ways that a chemist can use the atomic mass to figure out exactly how many atoms of something He's weighing out if he wants to do a particular chemical reaction. And this is all due to the fact that for every element, there's going to be some heavy members. Okay? And that's what those few extra digits past the decimal point are meant to indicate. Okay, so moving back. Okay, so let's talk a little bit about radioisotopes. Heavy isotopes will decompose to more stable forms because they have a lot of energy in their nucleus. They'll spontaneously decay and they'll release radioactivity, sort of like a tiny explosion, and this can transform that isotope into a different element. We can detect this energy with scanners. These are tools that can be used in biological research and medicine. They share the same chemistry as their normal isotopes, but because we have these scanners that can check where the energy is coming from, we can follow where the atoms go. And so we can build, for instance, radioactive versions of sugars or proteins, and we can feed them to living systems, and we can observe what happens to them. So this is an important diagnostic tool. It's also a way for us to look at living tissue for things like obstructions or openings. Barium, for instance, is a radioactive substance that we can use to run what we call GI series. We include a barium compound in sort of a thick milkshake consistency, and you drink that, and what it does is it coats your esophagus, your stomach, and your small and large intestine, and we can look for a block or we can look for a hole as a result of taking an image of the energy that's released from the radioactive isotope as it runs through the gastrointestinal system. So that's just one example. Another example is through PET scans, where we can feed people radioactive glucose and we can observe the brain using a scanner that can pick up where the radioactive sugar goes in the brain, and that'll tell us what parts of the brain are active and what parts of the brain are relatively quiet. All isotopes have the potential to damage living tissue. Some are used to destroy cancer, and other examples, such as radon from uranium decay, can actually cause cancer because it is a mutagen. Essentially, when any high energy source hits genetic material, which basically are the instructions that living cells use to dictate what they do and when they do it, we have the potential to change that information. If we change that information in a bad way, we can turn that cell from a normal cell into a cancer cell. And the problem, of course, with a cancer cell is it will begin to grow in an out of control fashion and steal nutrients and oxygen from the body and eventually kill the host if it's not destroyed first. Most atoms chemically combine with other atoms to form molecules and compounds. A molecule is basically what happens when two or more atoms are bound together. Examples include hydrogen gas, or in this case, is the formula for glucose. It's the smallest particle of a compound with the specific characteristics of that compound. So one molecule of glucose is going to have the same characteristics chemically as 20 pounds of glucose. A compound is where two or more different kinds of atoms are bound together. An example here would be glucose, but not hydrogen gas, because hydrogen gas is just two hydrogen atoms attached to each other, whereas glucose contains carbon, hydrogen, and oxygen, all bound to each other. Chemical bonds are essentially ways to attach atoms to each other. Electrons can occupy up to 7 electron shells around the nucleus, but it's the electrons in the outer shell that dictate the chemistry, and they also have the most energy. They're chemically reactive, and they obey something called the octet rule for the elements that we're interested in for the purposes of this course. And what the octet rule dictates is that except for the first shell around an atom, atoms will interact in such a way as to try to get 8 electrons in their outer shell. Now that might involve losing some electrons, that might involve stealing some electrons, or that might involve sharing some electrons. Okay? So here's a look at a couple of chemically inert elements. This is helium and neon. These are two members of the noble gas family. And what you can see is that both of these elements have full outer shells, so they don't need to gain or lose electrons or share electrons in order to exist at a low energy state. So they're perfectly happy, and that's why they're found in nature in their pure form. These guys don't have full valence shells. For instance, hydrogen can lose this electron, or it can gain another electron as it attempts to form um, molecules or compounds. Carbon has 4 electrons in its outer shell, so it can pick up another 4 in an attempt to form molecules or compounds. Now, the interesting thing, of course, about carbon is that carbon has the unique ability among all the elements in the periodic table to attach to itself as well as to other elements that are near it, that are near neighbors in the periodic table. So it can form rings and chains. And what's interesting about that is it provides a variety of shapes of different molecules, and that's important for living systems because shape will often dictate function. So this is why all life as we know it is based on carbon. Now notice that we can accommodate 4 more electrons in this outer shell. Carbon, as a result, can form 4 covalent bonds with other elements. Down here, sodium has 1 electron in its outer shell. So what it would like to do is kick this electron out, have this full outer shell be representative of a stable species, which in this case would be the sodium cation, and this electron would then go to some other element. So very often what will happen is that sodium likes to form salts with other compounds, and so it will generally take on a positive charge, while the species that picks up the electron will take on a negative charge, and they'll be attracted to each other as a result of the fact that opposite charges attract each other and charges that are similar to each other repel. Down here, oxygen has got 6 electrons in its outer shell, so it can pick up 2 more to fill. And so oxygen generally forms 2 covalent bonds. Every covalent bond represents a pair of electrons shared between 2 different elements. Now, in our discussion of the atom, you may have noticed something interesting about the atomic layout, which is that the electrons which orbit the nucleus have a negative charge, and we just said that negative charges repel each other, and that the nucleus has primarily positive charges, and we said that positive charges tend to repel each other. Well, why doesn't the atom then either collapse or explode? And that's a very good question, and the answer to that question is we're not entirely sure why the atom, as we understand it, remains a stable entity. This is part of the research that's going into particles that are even tinier than things like the protons, the neutrons, and the electrons, and what their role is in holding the atom together. It's something that we don't fully understand yet. So it's something to think about. It's an interesting question. If you take a physics course, they'll talk a lot about atomic structure and the forces that hold the atom together in a lot more detail than we'll address in this class. There's 3 types of chemical bonds: ionic, covalent, and hydrogen bonds. Ionic bonds are formed between 2 atoms that have opposite charges. The atoms will gain or lose electrons and as a result adapt a charge. The number of protons is not equal to the number of electrons in an ion. The transfer of valence shell electrons from one atom to another forms the ion. One will become the anion that has the negative charge. This will be an atom that has gained 1 or more electrons. And then one becomes a cation, which has a positive charge as a result of losing 1 or more electrons. The attraction of the opposite charges results in an ionic bond. And so here we can see an example of an ionic bond being formed. Remember that sodium has 1 electron in its outer shell, chlorine has 7, and so when these 2 elements get together, what sodium is going to try and do is kick this electron out, hand it over to chlorine, and look what happens as a result. The sodium takes on a positive charge, but it has a full outer shell, and that represents a low energy state for this atom. While the chlorine has picked up an extra electron and has a full outer shell, takes on a negative charge, and that represents a low energy state for that atom. And their attraction is the result of the interaction of their opposite charges. That's what holds them together. What's interesting about these two compounds is if you were to see pure sodium, it's very toxic. If pure sodium were to hit water, it would explode and burst into flames, whereas chlorine is an extremely corrosive, toxic gas. But when you react these two together, you get something that you put on your food all the time, which is good old table salt, sodium chloride. Most ionic compounds are salts. When dry salts form crystals instead of individual molecules, what we have is a stable arrangement of negative and positive particles. Examples shown here in sodium chloride. Notice the sodiums are indicated by the yellow balls and the chlorines by the green ones. And what we see is that every cation is surrounded by an anion, and every anion is surrounded on all sides by a cation. And the reason for that, again, is that it represents the low energy organization of those oppositely charged particles. So we've satisfied, again, the second law of thermodynamics here. Covalent bonds are formed by sharing two or more valence shell electrons, and this lets each atom fill its valence shell at least part of the time. And so this is an example of carbon reacting with hydrogen. Remember that carbon has 4 electrons in its outer shell. It can accommodate 4 more. So when it gets together with hydrogen, what happens is that it shares 4 electrons from its shell, and hydrogen shares 1 electron each from each of its shells. And the result is that you get a stable compound. And you know that because if you count the number of electrons in the outer shell, you find that the outer shell is full. So let's look at this element that we formed called methane. Notice right in the outer shell of carbon we have 2, 4, 6, 8 electrons. That represents a stable species. Let's look at hydrogen. 2 electrons in the outer shell here, 2 in the outer shell there, 2 here and 2 here. That represents stable electron arrangement for hydrogen. So what we have here is a stable compound because every element in the compound is has a full outer shell, and that represents a low energy situation and a stable compound. And so this is what the structural formula for methane would look like. If you're wondering, where do these electrons sit? Do they just kind of occupy this little physical space here? And the answer is no. What they do is that they travel between the atoms, right? So this pair of electrons would spend, sometime here and sometime there and sometime here and sometime there. Okay, so they go back and forth. Same with these guys, right? Sometime here and sometime there and sometime here and back and forth and back and forth. And the same is true here, okay? Back and forth and back and forth. And the same is true down here. At what speed do these electrons move? The speed of light. And in the bond shown here, the amount of time that the electron spends with the hydrogen and the amount of time that the electrons spend with the carbon are going to be about the same. And so that's what we call a nonpolar covalent bond because we're not generating in the molecule a separation of charge as a result of the electron spending more time with one atom than another. But sometimes what will happen is that we can share electrons unequally between atoms, and that results in what we call a polar covalent bond. And the consequence for the molecule is that it will have one part of the molecule that has a slight negative charge and the other part of the molecule that will have a slight positive charge. And the result there is that this will affect not only its chemistry but its behavior with other elements that are either polar or nonpolar. Bottom line, nonpolar things like to associate with nonpolar things because that doesn't require much energy, and polar things like to associate with polar things because that doesn't require much energy. On the next page we've got some examples of nonpolar covalent bonds. This is between 2 nitrogen atoms. Note nitrogen again has 5 electrons in the outer shell, so it can pick up 3. So when 2 nitrogen atoms get together, they form what we call a triple covalent bond, where they share 3 pair of electrons between the 2 nitrogen atoms that spend equal amounts of time with each atom, back and forth and back and forth. But again, full outer shells, right? 2, 4, 6, 8 in the outer shell here, 2, 4, 6, 8 in the outer shell here. Both atoms have full outer shells. Both atoms are low energy. As a result, we've created a stable situation. Down here with oxygen, we have 6 electrons in the outer shell, so we can share 2 pair between adjacent oxygen atoms, forming a double covalent bond. And if we look at the oxygen gas, what do we see? Well, 2, 4, 6, 2, 4, 6, 8 full outer shell here, and here 2, 4, 6, 8 full outer shell there. Both these oxygen atoms have a full outer shell. As a result, they're low energy. As a result, they are thermodynamically stable. Okay, so that's an example of nonpolar covalent bonding. The electrons are spending equal amounts of time with each atom. They're equally shared. Unequal sharing of electrons produces a dipole, basically a separation of charge on the molecule that causes it to behave differently than if the electrons were equally shared. The atoms in the bond have different electron attracting capabilities. The ability to attract an electron towards a particular atom is a property called electronegativity. Rule of thumb for electronegativity: basically anything to the right of carbon in the periodic table and to the left of the noble gases is an electronegative atom. The other atoms that we're going to deal with when we talk about biochemistry, carbon and hydrogen, those tend to be electroneutral. Okay, they tend to form nonpolar covalent bonds. Small atoms with 6 or 7 valence shell electrons are electronegative, examples being oxygen, nitrogen, sulfur, and so on. They have a strong ability to attract electrons. Most atoms with 1 or 2 valence shell electrons are electropositive. Those are metals. They tend to give up electrons. Examples would be things like sodium and lithium and so on. Here we see an example of a polar covalent bond. What's happening here is that the oxygen atom is bound to 2 hydrogen atoms. That forms a full outer shell for oxygen, okay, 8 in the outer shell, 2 in the outer shell for each hydrogen, but because the oxygen has a greater ability to pull the electrons towards it than the hydrogen does, this creates a negative region in the water molecule near the oxygen and a positive region near the hydrogen. And the result is that this is a polar molecule, meaning it likes to associate with other polar things. Now, how can you identify polar molecules? Well, for carbon-containing compounds, just look for electronegative atoms: oxygen, sulfur, nitrogen, and so on. If you see them, the likelihood is that that is a polar compound. Phosphorus is another example. Nonpolar compounds are not going to have those electronegative atoms. For biological molecules, they'll contain mostly carbon and hydrogen. And we'll see examples of each of these as we move through the rest of the talk. So here you can see an example of a comparison between the 3 types of bonding we've discussed: ionic, polar, and nonpolar covalent. In ionic bonds, you have a complete transfer of electrons, and as a result, the 2 charged particles are attracted to each other as a result of electrostatic forces. In a polar covalent bond, you have unequal sharing of electrons that produces a slight separation of charge within the molecule, but it's still a covalent bond. However, what that implicates for the molecule is that it's going to associate more easily with other polar compounds. And in a nonpolar covalent bond, we have equal sharing of electrons, so that the charge is balanced among atoms. You see this in all diatomic molecules like nitrogen gas, oxygen gas, hydrogen gas, and so on. But you also see it in hydrocarbons, where most of the covalent bonds are carbon-hydrogen bonds. That also represents essentially a nonpolar molecule. The attractive force between electropositive hydrogen and of one molecule and the electronegative atom of another molecule can produce some force known as a hydrogen bond. This is not a true bond. It's common between dipoles such as water, and it also acts as an intramolecular bond holding the large molecule in a 3-dimensional shape. So let's take a listen to hydrogen bonding.
The atoms that make up a water molecule are in a constant tug-of-war over their shared electrons. Oxygen exerts a far stronger pull on the shared electrons than does hydrogen, and so the electrons spend more of their time closer to the oxygen atom. Because of this unequal sharing of electrons, the oxygen atom in a water molecule actually has a slight negative charge, and each hydrogen atom has a slight positive charge, even though the water molecule as a whole is neutral. Because of the unequal sharing of electrons and the resulting positive and negative poles, a water molecule is said to be polar. The polarity of water molecules causes them to be attracted to each other. Since the positively charged atom involved in this special type of attraction is always a hydrogen atom, this kind of bond between molecules is called a hydrogen bond. Each water molecule can hydrogen bond to 4 other water molecules. A hydrogen bond is weak and lasts only a tiny fraction of a second, but it takes a lot of energy to overcome the combined attraction of many hydrogen bonds. This explains water's great capacity to store heat, its high boiling point, surface tension, and several other unusual properties.
This is an example of the attractive force of water for itself in action. This water strider is an insect that can walk across the surface of the water because the water is attracted more strongly to itself than it is to the air above it. This is also responsible for the ability of water to get to the tops of very tall trees. The leaves at the tops of the trees, well, really throughout the trees, have little structures on their undersides that are called stomates, that are like little pores that open up to the external air, and they allow the water to evaporate from the leaves. That produces a drawing force. In addition to that, the water is attracted to the cellulose, the wood that's in the plant. And because cellulose is a polar molecule and water is a polar molecule, that force, that polar interaction, which is the result of hydrogen bonding, as well as the pull from transpiration, gets water to the tops of tall trees so that they can have nourishment and they can continue to live. If water was not attracted to itself, plants would be limited to extremely short shrubs or structures that are very near the ground. Another example of this property of water is seen when you give blood. If you've ever had a blood donation, they give you a finger stick. You get that little drop of blood on the tip of the finger, and you put the little round glass tube onto that drop of blood. It'll wick up inside that glass tube. The glass is a polar compound. It's silicon dioxide. It's polar. It's attracted to the water in the blood plasma, which is polar, and that's what helps to pull the fluid up inside the capillary tube. Now, chemical reactions occur when chemical bonds are formed, rearranged, or broken, and they're represented as chemical equations using molecular formulas. The subscript indicates the atoms joined together by bonds, while the prefix denotes the number of unjoined atoms or molecules. Chemical equations contain reactants, which are the number and kind of reacting substances, and the chemical composition of the products, and the relative proportions of each reactant and product in the balanced equation. And so here's an example of a chemical reaction. Again, we have to obey the first law of thermodynamics, which states that what? Energy and matter can't be created or destroyed. So we have the equal number of atoms on the left side of the equation as we do on the right. And here you're seeing 2 hydrogen atoms reacting together to form hydrogen gas. Over here, we have 4 hydrogen atoms reacting with elemental carbon to form methane. This equation is balanced. As a result, if I were a chemist, I could use this to figure out how much of a particular reactant I would have to combine in a reaction vessel in order to get a particular amount of a compound. One of the ways that we balance equations is that we not only look at the subscripts, which are these little numbers down here, but we can also use coefficients, which are big numbers that would sit in front of a particular atom. One example is the chemical equation for respiration. So if I were to draw that in shorthand here, let's use black. Okay. Basically, the chemical formula for glucose is C6 H12, O6, and what happens is that this reacts with oxygen, O2, to produce carbon dioxide and water, CO2 plus H2O. This is what would happen, for instance, if you were to take a tablespoon of sugar and light it. Okay, you'd create heat and light, and you would generate carbon dioxide and water, and you would consume oxygen. But if you look at this equation, one of the things you notice is that it's not balanced, right? Because we have unequal numbers of carbon atoms on the left and right side, not to mention oxygen and hydrogen. So how do we balance this equation? Well, we can put coefficients here. If we put a 6 here, and we put a 6 there, and we put a 6 here, let's see what we come up with now. Let's count our oxygens. 2 times 6 is 12. That's 12 oxygen atoms plus 6 more is 18. How many oxygens we got here? 6 times 2 is 12 plus 6 more is 18. Oxygens are balanced. How about carbon? 6 carbons here, 6 carbons there. Check. And how about hydrogen? 6 times 2 is 12. And over here we've got 12 in the glucose, and so that's a balanced equation. Okay, so that's one of the ways that we use chemical equations to dictate how much of something we're going to mix with something else. Okay, some chemical reactions that we need to understand in living systems include synthesis reactions, decomposition, and exchange reactions. In a synthesis reaction, what happens is that 2 or more reactants combine to make one larger molecule. Okay, um, they always involve the formation of a novel bond, and they're said to be anabolic reactions. An example of a reaction like this would be the synthesis of a protein from its component amino acids, the production of a polysaccharide from monosaccharide subunits, or the building of DNA or RNA from their building blocks, which are called nucleotides. In order for these kind of reactions to work, energy has to be supplied on the left-hand side of the reaction to, to, to drive the generation of the product on the right side, because the product on the right side isn't just going to spontaneously form. And often this energy comes in the form of ATP, which, as we're going to find out, is the universal energy currency in living systems. Now, the counterpart to anabolic reactions are decomposition reactions, where we take a large molecule and we break it apart, and as we break those bonds, energy that gets released is transferred to usable energy in living systems in the form of ATP, which can drive other kinds of reactions, perhaps movement, perhaps synthesizing macromolecules. Any number of things can result from this form of readily available energy. Basically, as we break this molecule down into its tinier parts, as we break those chemical bonds, energy comes out, okay? And these are said to be catabolic reactions. And that energy can be used by the anabolic reactions to make macromolecules. It could be used by the cell to move or repair or grow, any number of things. In an exchange reaction, what happens is that um, an atom will switch what it's partnered with. And so you can see an example of an exchange reaction indicated up here. They're also called displacement reactions, and they involve both synthesis and decomposition. Bonds are both made and broken in exchange reactions. And the example we have here is of ATP transferring a phosphate group to a glucose molecule, forming glucose phosphate. The ATP is essentially releasing energy in this process, and that energy will go towards eventually extracting more chemical potential energy from the glucose molecule. Okay, but this is an example of an exchange reaction. And the bonds that you're seeing being made or broken in each of these are all covalent bonds. Okay. Um, Oxidation reduction reactions are decomposition reactions in which food fuels are broken down for energy. The oxidation is what sucks the energy out of the food. Reduction is the transfer of energy to a molecule. There are also exchange reactions because electrons are exchanged between the reactants. Electron donors lose electrons and are said to be oxidized, while the electron acceptors pick the electrons up and are said to be reduced. So as an example, we can see the equation that we just looked at a couple of slides ago. This is the equation for respiration, and you can see here that the glucose, which is here, is oxidized because the carbon now contains no more carbon-hydrogen bonds. It is also bound to more oxygen. That's one of the ways that you can tell it's been oxidized. Um, and the oxygen molecule is reduced because it picks up hydrogen bonds. So there's the oxygen and there's the reduced oxygen. Rule of thumb, in general, for oxidation reduction reactions in living systems, the more carbon-hydrogen bonds a molecule has, the more chemical potential energy it has, the more reduced it's said to be. The fewer carbon-hydrogen bonds an organic compound has, the more oxidized it is, the less energy it's said to have. Okay, so just kind of keep that in mind if you were to just look at a list of molecules on the board or on an exam or on a quiz. All chemical reactions either give off energy or consume energy. Exergonic reactions release energy. The products that result have less potential energy than the reactants do. Catabolic and oxidative reactions are responsible for these kinds of energy changes. Whereas endergonic reactions result in the net absorption of energy, the products have more potential energy than the reactants, and this involves primarily anabolic reactions. All chemical reactions are theoretically reversible, but not in practicality, because they all have to cross what we know as an energy barrier. Here's an example of a reaction going in a particular direction. A plus B makes AB. The reverse reaction is shown below it. A chemical equilibrium occurs if neither the forward nor reverse reaction is dominant, and this only happens when the energy barrier from the products to the reactants is very small. Many biological reactions are essentially irreversible because of the large energy barrier, and also due to the fact that often the products are removed, and so they can't participate in the reverse reaction anymore. The rate of chemical reactions is affected by the temperature. Basically, the higher the temperature, the higher the rate of the chemical reaction. The higher the concentration of the reaction, of the reactant, the greater the rate is going to be, and the smaller the particle size, the greater the rate is going to be. Now you might say, well, why is that? Well, let's kind of draw you, an example of a chemical reaction happening. So we got a little blue ball here, and we're going to have another, let's make a little red ball over here. And what happens when they collide is they form a product. Okay? So here we go. We have a chemical reaction, and now we make this new thing. Okay? So here's this new thing. There's our little red ball there. And then here's our little blue ball. Okay? In order for this thing to happen that we've just drawn here, these two things need to bump into each other. In order for them to bump into each other, they have to collide. So let's look at the first variable here. Increasing the temperature increases the rate. Well, why does that happen? Well, because the higher the temperature is, the more these things will move around. And the more they move around, the more likely they are to bump into each other and form a product. How about concentration? Well, here we've only drawn one red ball and one blue ball, but you could imagine if we had like 100 red balls and 100 blue balls and they were all moving around, that the rate at which they'd make their product would go way up because the number of collisions per unit time would go way up. Now, how about particle size? Well, the smaller something is, the easier it is to move around. The easier something is to move around, the more likely it is to collide with something else, and the result is more likely it is to produce a particular product. Okay? So that's the bottom line. That's why the rate of chemical reactions is affected the way it is. Now, there's one more variable that can affect the rate of a chemical reaction, and that's a catalyst. And what a catalyst does is it comes into a reaction and serves as a biological mediator. So essentially, in our description that we have up here now, if I were a catalyst, you could imagine me, let's just draw me as a pair of hands, okay? And so my pair of hands, I come into this reaction mess and I say, oh, I see you, okay? I'm going to grab you, and I see you over here, and I'm going to grab you over there. And when I grab you, I'm going to make you bump into each other in just the right orientation and with just enough energy to make this product over here. And then when I'm done, I'm going to move on to the next set of reactants. So I will disengage myself and I will move on to whatever it is I want to have react next. As a result, when I'm around, the rate of chemical reaction goes way up because the number of effective collisions goes up and the energy involved to make those collisions goes way down. In addition to that, you don't need a whole lot of catalyst to make a reaction speed up because the catalyst is not consumed in the reaction. Biological catalysts are called enzymes, which are proteins. They've evolved over thousands of years to help make chemical reactions in living systems happen without having to do things like crank up the temperature or mess too much with the concentrations of the reactants or the products. And that's important because living systems only tolerate a very narrow range of temperatures in order for their relevant chemical reactions to happen. And so the evolution of catalysts was a big deal for living systems because it allows the living system to make enough of what it needs quickly enough in order to continue to carry out life processes. Okay? All right. That brings me to the end of what I want to talk about in this segment. Stay tuned for part B of chapter 2. I'll see you in just a bit. Okay, welcome everyone. This is the back end of chapter 2. We're discussing chemistry, and we want to move into the area of the chemistry that specifically is found in processes important to life, and that's biochemistry. The study of chemical composition and reactions of living matter is defined as biochemistry, and all chemicals are either organic or inorganic. And what defines this, generally speaking, is the presence or absence of the element carbon. Okay, if you see carbon in the compound, you've got a pretty good idea that what you're dealing with is an organic compound, one that we would likely find in a living system. Inorganic compounds include molecules such as water, many salts, acids, and bases. They do not contain carbon. Organic compounds always contain carbon, and these include things like carbs and fats and proteins and the nucleic acids, DNA and RNA. They contain carbon, and they're usually covalently bonded, and they're essential for life. Now, there are a couple of exceptions to this carbon rule, and one is a molecule that we classify as inorganic, which is carbon dioxide. So this is the structure for carbon dioxide, CO2. And the reason that we classify this as an inorganic compound is because it doesn't contain any carbon-hydrogen bonds, and it's actually the metabolic end product of metabolism, which is a process whereby we extract chemical potential energy from macromolecules in the food that we eat, and we transfer it to a very important molecule that provides readily usable energy for living systems, which is called ATP. Okay, ATP, and we'll look at ATP a little bit more in detail a little bit later on in the talk. Now, one of the things that we have to understand about living systems is that in order for chemical reactions to happen, we have to have molecular movement, and that requires that we have a liquid environment, and that's one of the things that water provides for us. It's the most abundant inorganic compound, makes up about 70% of the volume of living cells, and some of its most important features that allow it to permit chemical reactions to occur are the amount of kinetic energy that's contained in water. Molecules can move in a liquid, they can bump into each other, and remember that one of the prerequisites for a chemical reaction is molecular collision. Other important properties we need to take note of is its high heat capacity. It absorbs and releases heat with relatively little temperature change, and this it helps to stabilize the temperature of living systems. Uh, to visualize this, think about what happens when, say, a lake freezes over. It'll transition from a liquid to a solid, but in the course of doing that, from going from a liquid to a solid, what it'll do is release heat to the surroundings, and this is called the heat of fusion, and that'll serve to warm up the air around it. By the same token, when it transitions from a solid to a liquid, what happens is that it absorbs a considerable amount of heat before it moves from a solid to a liquid. So what effect this has on living systems is that even though temperatures in the environment may fluctuate quite a bit, this high heat capacity of water keeps, for the human body, the temperature around 37 degrees centigrade. That heat that is released when water goes from a liquid to a gas is something else we need to acknowledge, and that's called the heat of vaporization. Okay? This evaporation requires a lot of heat, and this is one of the mechanisms the body uses to cool itself. For instance, on a hot day, when you're outside and you sweat, what happens is that the sweat will transition from a liquid to a gas, and that will remove heat from the skin surface. That'll cool the skin surface. That in turn will cool the blood that's at the skin surface, and that blood will in turn cool the organism. Okay? So the high heat capacity and the heat of vaporization, and also the fact, plain and simple, that it's a liquid. Now, another thing that we have to note about water is that it is polar, okay? Meaning that one end of the molecule has a slight negative charge and the other end of the molecule has a slight positive charge. And what this means in terms of interactions with solutes is that in order to, again, obey the second law of thermodynamics, the negative end of the molecule is going to seek out the positive end of the salt, and the positive end of the molecule is going to seek out the negative end of the salt. So if we imagine a salt crystal being dissolved in a beaker of water, as shown here, what will happen is that the water will be attracted to the edges of the crystal, and the oxygen will be attracted particularly to the sodium cations as the oxygen of the water inserts between the electrostatic attraction that exists between the chlorine and the salt. It'll break that electrostatic attraction up, and the salt crystal will begin to dissociate. Same thing is true with the partially positively charged hydrogens. They'll be attracted to the chloride anions. That will interfere with the interaction of the sodium cations and the chloride anions. The electrostatic attraction will be broken up, and the chloride anions will begin to solvate, dissociate into water. So slowly the edges of the crystal will fall away as the anions become hydrated, and eventually what will happen is the solution will go completely clear, and as a result you will have now basically a solution of ions. And this is also true for polar compounds like sugar. If you look at the chemical formula for glucose, C6H12O6, you have an organic molecule, but it has a lot of electronegative atoms in it. There's a lot of oxygens in that structural formula. And what that means is that the glucose is a polar molecule. It has slightly negative and slightly positive portions to the molecule, and what that means in terms of solvation is that the water can easily associate with it in a very similar way that it associates with the cations and anions of salts as shown here. Now, these layers that the water forms around these ions are called hydration layers. They're like large water cages, and what happens is that they help to sort of insulate the ions from interactions with other ions. This is what allows them to stay in solution. They can also form hydration layers around large charged molecules such as proteins, where they'll form what we know as colloids. An example of a colloid is something like Jello, okay, which is made entirely from protein as well. It's also an important transport medium in the body because it's capable of movement, and it also serves as a medium in which chemical reactions can take place. So this is one of the reasons why a majority of our body weight is actually water. Okay, so very important to understand water's properties and what water brings to the table. Now, water is often a necessary reactant in metabolic reactions. It's involved in hydrolysis and dehydration. In hydrolysis, of course, it's used to split macromolecules into their monomeric components, and in dehydration reactions, water is generated as it's removed when we assemble monomers into larger macromolecules. It also provides protection for certain organs from physical trauma. An example would be the cerebrospinal fluid that is produced by the ventricles in the brain and flows through hollow chambers both inside and outside the brainstem, and the spinal cord. And this allows the organ to float in a liquid medium without coming into contact with the bones that surround these structures. So here you can see again the protective aspect of water as well. So let's talk a little bit about the importance of electrolytes. Electrolytes are vital because they can influence where water goes in the body. They also serve as conductors of electrical signals, and they also participate as cofactors in a lot of enzymatic reactions. They help certain enzymes allow their reactants to be transformed into products. Um, they also serve as transport. They serve to aid in transport. An example would be the iron that you find in hemoglobin, which is used as a binding site for oxygen in the context of the hemoglobin molecule. And they also are important for fluid balance in the body as a result of their influence over osmosis. They contain cations other than protons and anions other than hydroxide anions. Some common salts in the body include sodium chloride, calcium carbonate, potassium chloride, and certain other calcium phosphates. So this is one of the reasons why, and again, most salts you would define as trace elements. But remember, just because they're present in small amounts in the body doesn't mean that they're unimportant. Without these vital roles, these processes would shut down. If we couldn't send and receive electrical signals in the body, we'd very rapidly die because the musculoskeletal system and the nervous system would cease to function. If we couldn't conduct oxygen in the body and its role as a transport facilitator, then we wouldn't be able to generate enough energy in the cells to sustain life. And if we didn't have these salts in the body, we wouldn't be able to influence where body water moves. And as a result, we would suffer from things like hypotonic hydration or dehydration. So this is, again, one of the body's main mechanisms to maintain our fluid balance. Okay, now one of the species that are important in chemical reactions are acids and bases. Now they're both electrolytes because they dissociate in water, but acids are defined as proton donors. And what we can see here is an example of a strong acid, hydrochloric acid, which is the acid that's found in the stomach, in water will almost completely dissociate into free protons and chloride anions. And what this will do is this will lower the pH of the solution in which it finds itself. Okay? So as acidity increases, pH decreases. Bases, on the other hand, are proton acceptors. Now this is an example of a strong base. This is Li. Okay? And what you see is that it readily dissociates into cations and hydroxide anions. And what happens with the hydroxide anion is that it will readily pick up free protons from water, because water ionizes at a very low rate all by itself. A glass of distilled water will undergo the following equilibrium reaction. If we draw it here, H2O will ionize to a very small extent into protons and hydroxide anions. And it does that at a rate of 1 times 10 to the minus 7th molecules per liter of solution. And this is why distilled water has a pH of 7, which is defined as neutrality. Okay? So there's the autoionization of water. And in a strong base, what happens is that the hydroxide anion will intercept these free protons. It'll grab these guys, okay, and incorporate them into a molecule of water. And what that'll do is effectively reduce the free proton concentration, and what that causes to happen is for the pH to rise. Okay, so as basicity increases, the pH goes up. Some important physiological acids include hydrochloric acid, found in the stomach, acetic acid, which is a byproduct of a lot of metabolic reactions, and carbonic acid, which is a physiological buffer system that we see in body fluids and in plasma. Important physiological bases include bicarbonate, which is also part of the carbonic acid bicarbonate buffer system, and ammonia, which is a byproduct of nucleic acid and protein breakdown. Now, you can see why these acids are acids, right? You can see the hydrogens in each of these species that will dissociate in water from each of these compounds, and as a result decrease the pH. It's not quite as obvious with ammonia and bicarbonate. Bicarbonate, shown here, will pick up a proton from the autoionization of water and form carbonic acid. Okay, so it will end up generating now this molecule over here, okay, after it picks up a proton. Ammonia might be a little bit more difficult to figure out why it's a base. Let's draw ammonia. Let's draw the chemical structure of ammonia, which is NH3. Remember that ammonia has, or that nitrogen has 5 electrons in its outer shell, which means that it can form 3 covalent bonds, because that will complete the outer octet of 8 electrons in that outer shell. But what this means for ammonia is that there's a pair of electrons that are not going to be involved in a chemical bond, and what happens due to the autoionization of water is that this ammonia can pick up a proton, okay, and can end up forming ammonium, which was NH4, where the positive charge will reside on the nitrogen. And what that does, again, to the pH is it causes it to rise. Okay? So most nitrogen-containing compounds are considered to be bases. So when you hear about nitrogenous bases, say in nucleotides, or when you hear about certain amino acid residues being basic, what we mean generally is that they contain nitrogen, and that lone pair of electrons acts as a proton scavenger, and that's what causes the pH to go up. Now, what is pH? We've been talking about it for a few minutes now, and it's basically a measurement of the amount of free protons in a system. The system would be a certain solution. Basically, this little p here has a definite mathematical definition. So let's draw what little p literally means. We'll do this down here in red. When we say pH, okay, what we're saying mathematically is that we're taking a quantity, which is the opposite of the log of the proton concentration measured as a molar unit. Okay? So that's literally what pH means. So how does this work in reality when we're doing computations? Well, let's say that we had a solution that had a molar concentration of protons of 10 to the minus 7th molar. Now remember that when we say molar, okay, 10 to the minus 7th molar, that's the same thing as saying 10 to the minus 7th moles per liter. Per liter, right there, okay? So if we substitute this quantity back into this equation, what we end up with is that the pH would equal the opposite of the log of 10 to the minus 7th. And what that computes out to is the number 7. And you can do this in your calculator to confirm what I've told you. Okay? So that's what pH literally means. Little operator p means the opposite of the log to the base 10 of whatever comes after p. Okay? So it's a measurement of the amount of free protons in solution. As the amount of free protons increases, what happens is the acidity goes up and the basicity goes down. The concentration of hydroxide anions decreases as the concentration of protons goes up. The pH begins to drop. On the other hand, if we look at basicity, as the concentration of free protons decreases, the basicity or the alkalinity goes up and the acidity goes down, and the result is that the pH will rise. Okay, so as hydroxide anions increase, as the proton concentration decreases. pH is measured as the negative logarithm of the concentration of protons in molar measurement, and the scale ranges from 0 to 14. Because the pH scale is a log scale, what that means is for every unit in pH change, you're looking at a tenfold difference in concentration. So as an example, something that is pH 7 is going to have 100 times more protons than something that is pH 9. All right, so you can work that out using the formula, and you'll see that that is in fact correct. Now, one of the useful equations that you can employ to compute these ratios of ions is a relatively easy one, and what it states is that in any aqueous solution, the pH of the solution plus the pOH of the solution, which is the opposite of the log of the hydroxide ion concentration, is equal to 14. Okay, equals 14. So if the pH of a solution is 7, the pOH is also 7. If the pH of a solution is 8, the pOH of the solution is 6, and so on. Okay, so useful equation up there. Kind of commit that to memory. Okay, this is the pH scale, and what it's showing you are the proton concentrations of some standard solutions that you come into contact with all your life. Remember that as the proton concentration goes up, the pH drops. Acidic pH is anything below 7. Neutrality is defined as 7, and then basic is anything that is higher than 7. Okay? So pure water right here has a pH of exactly 7 because that's its ionization ratio. And you can see here some examples of some common bases above the neutrality area on the pH scale. Sodium hydroxide, which you know better as oven cleaner, pH 14. Household ammonia around here, around pH 11. Bleach around pH 9 or so. Egg whites, pH 8. Blood, slightly alkaline at pH 7.4. Milk, oddly enough, slightly acidic, right? Around 6.3. Black coffee, pH 5. Wine, around pH 3. Lemon juice, around pH 2. And hydrochloric acid, concentrated, pH 0. Okay, so we talked about the fact. Let's look at hydrochloric acid just for a second here, because this is kind of interesting. That hydrochloric acid is also known as stomach acid, and because the pH is so low, it's an extremely corrosive solution, meaning that it will degrade biological material that it comes into contact with. And so you might well ask the question, oh my goodness, why doesn't the stomach just digest itself as a result of all that acid that's in it? And the answer to your question is that the stomach does digest itself, but because of several factors. Number one, the fact that the stomach lining is coated with an alkaline mucus that protects the lining from the acidic secretions. And number two, the fact that the stomach rebuilds its lining faster than it digests it. That maintains the integrity of the stomach as a digestive organ. Okay, so the stomach is uniquely suited to this kind of environment. If you look at the pH of the stomach, it is in fact around pH 2, okay? And the reason for that is that the stomach acid is buffered by components that are in the gastric juice other than just straight hydrochloric acid. And the purpose of the hydrochloric acid is to hydrolyze the macromolecules that come into the stomach from the esophagus, which helps chemically break down the food that we eat, so that eventually, as it passes through the small intestine, we can absorb it into the bloodstream and then transport it from there to the liver, which will then either store it for later use or will pass it on to the rest of the body to use as fuel. Okay, neutralization results from the mixing of acids and bases. What usually happens when you mix an acid and a base is you'll get a chemical reaction. It may foam, it may give off some gas, and what will happen is a displacement reaction that will generate a water and a salt. Okay, as an example, if we were to take, now don't try this at home, if we were to take sodium hydroxide, okay, NaOH, and we were to mix it with hydrochloric acid, HCl, and we were to do this in water, okay, what we would generate, number one, we would generate a lot of heat, okay, so heat is represented here by a triangle, and there would be some fizzing, so fizz, and there would be a release of a little bit of gas, and at the end of the day, what we would have is sodium chloride and water. So we would end up with basically a salt solution. Okay, and that's an example of a neutralization reaction. If we had the exact ratios of sodium hydroxide to hydrochloric acid, if we mixed them 1-to-1 like the equation shows, when we're done with this neutralization reaction, the pH on this side of the equation would be 7, okay, once the reaction is finished. In the body, acids and bases help maintain homeostasis. pH changes will interfere with cell function and can damage living tissue. So the body likes to keep most living tissues around pH 7 or so. And so what it'll do in order to avoid um, the pH becoming too acidic or too basic is it will use buffer systems and also physiological processes to try and maintain the pH of the aqueous environment of the cell and outside the cell close to 7. A tiny change in pH can be fatal, and pH is regulated by physiological buffer systems as well as the urinary system, the respiratory system, all acting together in concert. Now, you might wonder, well, how does this work? Okay, so we're not really to the respiratory system or the urinary system yet, but I'll just give you kind of an overview here. One of the things that happens sometimes in the body is that the pH can momentarily drop. Okay, so let's say that your body's pH drops a slight bit, and this is often due to the accumulation of carbon dioxide in plasma and in interstitial fluid. So A reduction in pH is essentially caused forgive my writing here, I'm using a mouse pad, caused by an increase in CO2 in solution. Now, why does this happen? Well, because as CO2 builds up, it will combine with water and it'll form carbonic acid, H2CO3. The carbonic acid, in turn, it will dissociate and form free protons and bicarbonate, HCO3 minus. Sorry, I didn't mean to write that there. Ignore that arrow. Plus HCO3 minus. Now, these reactions are in equilibrium, okay, so we can go back and forth here, but the more carbon dioxide we build up in the fluids, the more we're going to push this equilibrium in the downward direction, as pictured here. And as the free protons begin to accumulate, the pH begins to drop. So how does the respiratory system deal with that? Well, what will happen is that chemoreceptors, which are special um, cells in the nervous system that can basically test the chemical composition of blood plasma and tissue fluid, will detect that the pH is beginning to drop, and they will send electrical signals to the brain that will cause the respiratory rate to increase. As the respiratory rate goes up, you're going to blow off more carbon dioxide as you breathe out. What that'll do is that'll shift this equilibrium back in the opposite direction. When that happens, protons will be consumed and the pH will begin to rise back towards normal. Okay, so that's how the respiratory system helps to regulate pH. Buffers, as you can see here, are designed to modify pH by converting strong acids to weak ones or strong bases to weak ones, and we'll see in a minute exactly how that works. How about the urinary system, the kidneys? How do they regulate pH? Well, what they can do is, depending on the situation in the body, they can either secrete acidic urine or basic urine. So they can secrete protons or bicarbonate, and the result then is to shift the pH back towards 7.4, depending on the nature of the pH change. Okay, so that's how the lungs and the kidneys work. All right. So the next thing we want to touch on are buffers. How do buffers mediate pH in aqueous systems? Remember that acidity is a measurement directly of free protons in solution that are not bound to anions. Buffers will resist changes in pH by releasing hydrogen ions if the pH goes up, or by binding hydrogen ions if the pH falls. So they convert strong acids or bases into weak ones. An example is the carbonic acid bicarbonate system that's shown here. They can shift back and forth. So let's imagine this system for a minute. Okay, we're going to do some sort of virtual experiments here. And let's say that these three components are in a beaker in water. Okay, so there's our beaker. Okay, and there's our solution. And these three components are already here waiting, all right, for, in this case, a challenge. So let's say that we challenge this solution with a strong acid. So let's say we throw some hydrochloric acid in here. Now, what do we know about strong acids? Strong acids are going to dissociate very, very rapidly, okay? So the hydrochloric acid will quickly dissociate into protons and chloride anions. That's what a strong acid does. That's the definition of a strong acid. Okay, complete dissociation and massive production of free protons. Now, in the buffered solution, though, we have a trick up our sleeve, and the trick up our sleeve is simply this: that the bicarbonate can intercept these protons and convert back into carbonic acid. And the definition of a weak acid is one that does not dissociate to a great extent. And so what we've done here is we've locked up these protons before they get a chance to drop the pH significantly by now caging them in the context of a weak acid. All right, now let's look at what happens if we have a base challenge. So let's take our same situation. Let me flip forward and back here real quick. Let's take our same situation, and we'll draw our beaker. Okay. And there's our water. And we've got this buffer system still in the beaker. And now instead of challenging it with a strong acid, we're going to challenge it with sodium hydroxide, a very strong base. So what do we know about sodium hydroxide? What we know is that it will readily dissociate into sodium and hydroxide. Okay. Now, if this were an unbuffered solution, the pH would rise dramatically. But we have a buffer system here. So there's the sodium, there's the hydroxide. It's the hydroxide that's going to try and steal protons from water, and that will result now in an increase in pH. So let's put the plus there, and then let's put the minus over here. Okay. But what happens? Well, we have another trick up our sleeve, right? And what's the trick? Well, this time what comes to the rescue is the carbonic acid. A strong base can steal a proton from a weak acid. And when that happens, what will occur is that the hydroxide, okay, as it meets with the carbonic acid, will form water, and the carbonic acid, which has had its proton stolen from it, will form now bicarbonate. Okay, so what have we done? We have now turned the strong base, the sodium hydroxide, into a weak base, the bicarbonate, and the result is that the pH shift is going to be minimal. Okay, so that's how these buffer systems work. There's a lot of other buffer systems around. The bicarbonate buffer system is just one of many. Proteins can serve as buffer systems. Phosphoric acid can serve as a buffer system, and they all work in a very similar fashion. Basically, they play ping pong with the protons so that we don't have a significant pH shift when we have an acid or base challenge. So you can see how in the human body, with the urinary system, the respiratory system, and these biological buffers, we tend to keep pH pretty close to 7.4, with a few exceptions. The few exceptions being places like the stomach and in certain digestive organs such as the pancreas, where the pH might be slightly basic, okay, as a result of the production of bicarbonate in pancreatic juice. But generally, everywhere you look in the body, the pH is going to be right around 7.4, and it's for the reasons that we've detailed here, right? These buffer systems and the urinary system and respiratory system all acting together. Okay, so what we want to do next is take a tour of some of the organic compounds that are out there. These are molecules that contain carbon, with the exception of carbon dioxide, as we discussed earlier, and carbon monoxide, which is actually a metabolic poison. And the reason we don't consider those organic compounds is because they don't contain carbon-hydrogen bonds. Carbon is considered to be electroneutral, meaning that it doesn't, when it binds, particularly with hydrogen, it doesn't pull electrons towards it more than the hydrogen atom does. Okay, so when you see carbon-hydrogen bonds, you can think of them basically as nonpolar covalent bonds. The other thing, of course, about carbon, remember, is that carbon has 4 electrons in its outer shell. Whoops. 4 electrons in its outer shell, which means that it can form 4 covalent bonds. All right, carbon's atomic number is 6. There's 2 electrons in the first shell, and there are 4 electrons in the valence shell. This is unique to living systems. The nice thing about carbon, of course, is that it has the unique ability to form rings and chains, and as a result, you can build a variety of different shaped molecules that can perform a variety of different functions, and very few elements in the periodic table can do that. The major organic compounds we're going to discuss are carbohydrates, lipids, proteins, and nucleic acids. A lot of these are polymers, which means that they're built up from chains of simpler molecules to make macromolecules. They're synthesized primarily by dehydration synthesis, which means that we remove water in order to form the macromolecule, and they're broken down by a process known as hydrolysis, where we use water to actually split the molecule. And so you can see examples of each of these here. This is sort of a generic dehydration synthesis reaction. Water is removed, and we now build a polymer of 2 monomers. This would be considered an anabolic reaction, meaning we're making big molecules from tiny building blocks. And hydrolysis is shown below, okay, where we take water. Hydrolysis literally means using water to split. The water is incorporated into this chemical bond, and what happens now is that we free the monomers from each other, and generally those are what we call catabolic reactions, where we're breaking apart a macromolecule, primarily in most cases for the purposes of extracting energy from it. Here's an example of an actual dehydration and synthesis and hydrolysis reaction. Here you see the formation of sucrose. Glucose and fructose are the monosaccharides. Glucose has a 6-membered ring and fructose has a 5-membered ring, with oxygen occupying one of the points in the ring. And what happens in the formation of sucrose is that we remove a water molecule and we form now a unique bond called a glycosidic bond between the glucose and the fructose. And this would again be an anabolic reaction. In the opposite direction, if we wanted to free these monomers from each other, we would use water to split this chemical bond and generate the 2 monosaccharides, fructose and glucose, for the purposes, in most cases, of generating energy for the cell. So let's talk a little bit about carbohydrates. Carbohydrates are important primarily as an energy source. They're one of the 3 macronutrients, the macronutrients being proteins, fats, and carbohydrates. They can either be in the form of simple sugars like monosaccharides, or they can be chains of simple sugars. If it's 2 sugars, it's considered a disaccharide, 3 sugars, a trisaccharide. If it's more than 3, we consider it to be a polysaccharide. An example of a polysaccharide that's a common component in your diet is starch, which is a glucose polymer. Carbohydrates contain carbon, hydrogen, and oxygen in this ratio, double the amount of hydrogens as there are carbons and oxygen. So if you're confused as to whether or not a compound is a fat, is a carbohydrate, or is a protein, always look for this ratio, okay? There's 3 classes: monosaccharides, single sugars; disaccharides 2; polysaccharides, generally more than 3. And they have a lot of different functions. They are, of course, a fuel, but they also have a structural role. For instance, they're found in the building blocks of the information molecules DNA and RNA. Ribose is the 5-membered ring sugar that is part of the building blocks of ribonucleotides, which are what we make RNA out of. And deoxyribose is a sugar that is the part of the backbone of deoxyribonucleotides, which are the building blocks of DNA. Okay. Um, Other important functions of carbohydrates is they serve as identification molecules on the surfaces of cell membranes. This is for the immune system to be able to identify cells that belong inside the organism versus cells that don't. They also serve as um, essentially molecule tags. One of the ways that we direct proteins to certain destinations inside the cell is by putting carbohydrate residues on certain proteins as address labels, and then a very special cellular organelle called the Golgi apparatus will then box those proteins up and send them to their final destination in the cell, largely due to having that proper collection of carbohydrates on the cell surface. They're also important as a component of connective tissue. They're part of what we know as the extracellular matrix, which is a product of connective tissue that's used to connect tissue types to each other, kind of like the body's glue. Okay, so those are just a few of the functions of carbohydrates. Okay, so what we want to touch on next are monosaccharides, and these are simple sugars that have 7 to 3 carbon atoms. The general formula is given to the left, CH2O. Remember that you have generally double the amount of hydrogens as you do oxygens or carbons in these molecules, and because of the high number of oxygen atoms you have proportionally in the molecule, it tends to be polar, so it dissolves very well in water. These are monomers of carbohydrates. Important monosaccharides include the pentose sugars ribose and deoxyribose, which are structural components of nucleotides, which are the building blocks of RNA and DNA. Hexose sugars, such as glucose, which are important in the generation of energy for cells. Okay? And you can see examples again below of each of these molecules. Now, disaccharides are double sugars that are too large to pass through cell membranes, so they have to be hydrolytically processed in order for them to be absorbed across the lining of the digestive tract and into the blood. Important disaccharides include sucrose, maltose, and lactose. And what we're going to do is watch a little video here that talks a little bit about where disaccharides come from. So take a listen. Okay, now polysaccharides are simply monomers or polymers of monosaccharides. The monosaccharides would serve as the links in what the polysaccharide would be the chain of. Important polysaccharides include starch, which is a major foodstuff, and glycogen, which is also called animal starch and is produced in human beings in liver and muscle. It tends not to be very soluble, so when we find starch inside living cells, it tends to be stored as granules or inclusion bodies and serves as a source of quick energy for cells that sometimes need to fill an energy deficit due to temporary lack of oxygen or nutrients coming from the bloodstream. So let's take a look at how polysaccharides are formed. Okay, lipids are the most energy-rich, or another way to put it would be calorie-rich molecule in living systems. They contain more energy than any other macronutrient, more than carbohydrates and more than proteins. And the reason for this is that they contain a lot of carbon-hydrogen bonds, as you can see in the diagram here. Essentially, a way to rank the energy content of organic molecules for living systems is to count the number of carbon-hydrogen bonds. The more carbon-hydrogen bonds you count, the higher the energy content is going to be. And so you can see here that we have considerably more carbon-hydrogen bonds than in a polysaccharide of equivalent molecular weight or a protein of equivalent molecular weight. Now, sometimes you'll find phosphorus atoms associated with these lipids, particularly when they form structural components of membranes known as phospholipids. they tend to be insoluble in water, and this is due to the fact that large sections of the molecule are hydrophobic. And what we mean by hydrophobic is they don't have a significant separation of charge in the molecule due to a lack of electronegative atoms. And if you look at the molecule here, that's exactly what you see, is that in this region, which I'll circle in red, we tend to find relatively few oxygens, nitrogens, sulfurs, and so on. Mostly carbon and hydrogen here. So you could think of this part of the molecule as greasy. Okay? And what do we know about how the second law of thermodynamics affects the way these molecules interact with water? Well, remember that we talked about the fact that the second law of thermodynamics dictates that molecules of similar chemical characteristics will associate with each other because that requires the least amount of energy. And in the case of hydrophobic molecules, what that entails is that the hydrophobic part of the lipid is going to interact with other hydrophobic portions of lipids or other hydrophobic molecules because that requires the least energy to maintain. The lipid shown here is a triglyceride. Excuse me. Also called a neutral fat. It's used as starting material for the production of phospholipids. Other types of relevant lipids include steroids and eicosanoids. And so let's take a listen to how it is that triglycerides are formed and what roles lipids have in the body.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules.
Okay, now triglycerides or neutral fats are molecules that are built out of a glycerol backbone and the dehydration synthesis that attaches 3 fatty acid molecules to that backbone. They're termed fats when they're solid at room temperature and oils when they're liquid at room temperature. And basically what determines whether it's a fat, whether it's a fat or an oil is the length of the fatty acid chain and the degree of unsaturation, meaning the number of double bonds that you would find in any of these 3 tails that are hanging off of the glycerol. Essentially, the shorter the fatty acid tail, the more likely it is to be an oil, and the higher the degree of unsaturation, the more likely it is to be an oil. When the chains are long and saturated, this tends to be a solid at room temperature. The main functions are energy storage, insulation, and protection. Now, saturated fatty acids have only single covalent bonds between their carbons, meaning they're bound to the maximum amount of hydrogen they can possibly attach to. And what this means is that they contain the maximum amount of potential calories for living systems. Solid animal fats such as butter are examples of saturated fatty acids. Unsaturated fatty acids have 1 or more double bonds between the carbon atoms. As a result, they have a lower calorie content and they have less chemical potential energy than their saturated counterparts. Examples include plant oils such as olive oil. These tend to be heart healthy because they contain fewer calories. They're also less likely to contribute into the body to species in blood plasma that we call bad cholesterol, which are simply combinations of proteins, triglycerides, and cholesterol in the body that travel through the plasma that act as vehicles to move these lipids from point A to point B inside a living system. Essentially, when we have a lot of saturated fatty acids in our diet, we tend to make a lot more of what we call these bad cholesterols, which are these chylomicrons and these very low density lipoproteins and low density lipoproteins. And what they tend to do is to accumulate on the walls of arteries. And what this causes to happen is for the arteries to narrow in diameter and become stiffer. And that slows blood flow through those areas. And one of the things that you'll learn about blood when we get to the circulatory system is that the slower blood flows, the more likely it is to clot. And this can lead to increased risk of heart attack and stroke. In addition to that, what will happen as the narrow arteries become more pervasive is that the blood pressure will go up because the force of the blood against the walls of the vessel is going to increase as the volume of the vessel decreases. It's kind of like squeezing a water balloon till it pops. And of course, this puts a huge strain on the heart and causes you to run increased risk of myocardial infarction. Omega-3 fatty acids are examples of trans fats. Omega-3 fatty acids are an example of these heart-healthy fatty acids. Trans fats are oils that were at one point, fatty acids rather, that were at one point polyunsaturated or unsaturated, and they've been chemically modified to contain more carbon-hydrogen bonds. So they have, as a result, a higher calorie content. The reason we try to avoid trans fats in our diet is because they can contribute to this problem of arteriosclerosis, hypertension, and increased risk of heart attack and stroke that we associate with the formation of these bad types of cholesterol, these VLDLs and LDLs and chylomicrons. The reason that trans fats are used in a lot of snack products is because they're less likely to spoil and go rancid, and as a result, the product has a longer shelf life. So it's not a plot by companies to try and make you unhealthy. It's simply a way for their product to be able to be viable on the shelf longer so that they can sell more product, make more money, hire more people, and create more jobs. Okay, phospholipids are modified triglycerides. Glycerol plus two fatty acid groups is attached at its opposite end to a head region that contains a phosphate group and often, very frequently, some kind of amine, okay, which is a nitrogen-containing compound that may be attached to one or more methyl groups. This is important in cell membrane structure because what this provides is something that we call an amphiphatic molecule, where if we observe, let's use blue here, this part of the molecule that I'm circling has a polar or charged aspect to it, depending on the groups that are attached to the phosphate. You can see here in this example, the nitrogen has a positive charge and the phosphorus has a negative charge. The bottom line is that this portion of the molecule is going to tend to associate with other polar molecules, most importantly water, due to the fact that it itself has a charged or polar nature. Same thing is true of the glycerol backbone. It has quite a few electronegative atoms attached to it, and so this part of the molecule we call the polar head of the phospholipid. And then this part that I'll circle in red is, well, let's circle it in black, is the nonpolar portion. And as you can see, it's dominated by carbon-hydrogen bonds. And this part of the molecule would be what you could think of as the greasy part of the molecule. And so what they've done here is they've shown you an example of this molecule in its sort of a diagrammatic form, showing you the polar heads and the nonpolar tails in the context of a cell membrane, which is the protective semi-permeable outer layer of living cells. The arrangement of these phospholipids is such that the polar heads tend to project towards the water or the cytoplasm, and the nonpolar tails tend to project into the center of this lipid bilayer, and what this produces is a semi-permeable barrier around the membrane that maintains this arrangement, again, due to the fact that it's energetically favorable. Notice that the nonpolar stuff associates with the nonpolar stuff, and the polar stuff associates with water or cytoplasm. So let's draw that in real quick. This would be the water here, H2O, right, on the outside of the cell. And then on the other side of the cell, you would have cytoplasm, which is mostly water. So CYT for short. And so what this means is that molecules that are polar or charged can only get into the cell through special channels, which are provided by membrane proteins, integral proteins that essentially allow a hole to exist through the plasma membrane that the cell can open or shut depending on its need for that particular compound. Nonpolar components, such as steroids or other lipid-soluble molecules, can slide very easily through the lipid bilayer because the hydrophobic middle doesn't present much of an energy barrier to it. So something like a steroid, let's just ask for steroid, can really just sort of slide right through here, right between the phospholipids, and go straight into the cytoplasm and exert its effects on the cell. Okay, so that's the significance of the phospholipid, and we'll touch on that a little bit more when we get to the chapter on cell biology. Okay, steroids are a lipid that's made up of 4 interlocking rings. You can see here 3 6-membered rings fused together, sharing a side, and 1 5-membered ring. Cholesterol is the starting point for most steroids. It also serves as the building block for vitamin D, steroid hormones, and bile salts. They all have the same basic structure. And what you notice about this structure is that it's mostly carbon-hydrogen. Now there is a few electronegative atoms that you can find if you look through things like vitamin D and steroid hormones, but they don't make up the majority of the molecule. So you could think of this entire thing, let's just circle the part that we would consider greasy, okay? This whole thing here is relatively lipid soluble, okay? It likes to associate with other molecules that are also nonpolar. That's why it can slide through cell membranes so easily. The most important steroid is cholesterol because it's the building block of a lot of these other compounds. We find it as a portion of cell membranes where it controls membrane fluidity. You could think of cholesterol molecules in the cell membrane as acting a little bit like door stops that keep a door from swinging back and forth. In the case of the cell membrane, the cholesterol acts as a doorstop to keep the phospholipids from moving significantly either forward or back or left or right. They are able to move in the fluid mosaic model in a two-dimensional plane. And so the more cholesterol that's in a cell membrane, the less fluid it is. The less cholesterol in the cell membrane, the more fluid it is. And the cell can control how much cholesterol it wants to stick in the membrane, and that'll affect the movement not just of the phospholipids, but also of other molecules that are in the cell membrane, like integral membrane proteins, cytoskeletal elements, and so forth. It also serves as a building block for vitamin D, steroid hormones, and bile salts, and we'll encounter each of these as we travel through the different body systems. Eicosanoids are derivatives of fatty acids. There are a lot of different types of eicosanoids. Most are derived from arachidonic acid that's a component of cell membranes. The most important eicosanoids are prostaglandins, which are important in blood clotting, controlling blood pressure, inflammation, and labor contractions. Now, some of these you might have some familiarity with. Blood pressure is simply the force of blood against the walls of the vessels. Okay, somebody who has high blood pressure is said to be hypertensive. Somebody that has low blood pressure is said to have hypotension. Inflammation is essentially a process that the body uses in the face of injury to avoid infection and disease and to eventually engage in tissue repair. And essentially what inflammation consists of is the area of the injury turning red, swelling up, hurting, and increasing in temperature. And what those responses are all designed to do is to bring blood to the area. The blood contains white blood cells, which are germ fighters, and the blood also brings with it eventually cells that will um, produce a clot, and also after the clot's no longer needed, or once the tissue's been repaired, will dissolve that clot. So this is the purpose for inflammation. All the components are important, right? The redness is because the blood comes to the area. The swelling is because of the collection of fluid. The pain is because the pressure presses on nerve endings that make pain, and that tells us not to use the affected area. And the increase in temperature is designed to allow the white blood cells to reproduce more quickly than the pathogen does, and that's what helps us again stem the tide of potential infection, because every injury represents a potential port of entry for a pathogen inside the body. They're also important in labor contractions. This is the smooth muscle layer in the uterus, which is what contains the developing baby for 9 months. And when the baby is going to be expelled, what happens is that prostaglandins, in part, are responsible for initiating these smooth muscle contractions that help to force the baby out head first through the birth canal so that the baby can be delivered. Other lipids in the body include the fat-soluble vitamins A, D, E, and K. Now, we have to be very careful about taking these vitamins because they do dissolve in fat, and as a result, we can easily overdose on any of these vitamins. But they're all very important. Vitamin K is critical in blood clotting, vitamin E important for wound healing, vitamin D important for the mineralization of bone, and vitamin A is a major component in retinol, which is the visual pigment that's found in the back of the eyeball, which is along with its associated protein rhodopsin, responsible for generating electrical currents in response to light that comes into the eye. So this is vitamin A is part of what makes us able to see. And we get most of our vitamin A from our yellow and our orange vegetables. So that's why Mom always told you to eat your carrots so you could see well at night. She wasn't lying. Lipoproteins are important in transporting fats in the blood. And what lipoproteins do is they serve as a vehicle to move these lipids around in a system that's primarily made of water. Most of the plasmid is made up of water, and as a result, it's a polar solution. So in order to transport something that's hydrophobic in water, you have to enclose it in, in a molecule that is hydrophobic on one side and hydrophilic on the other, and that's what the lipoproteins allow. Essentially, you could think of them as little taxi cabs that move lipids around the body from place to place. We couldn't just put those lipids directly into the plasma because they wouldn't dissolve effectively, and as a result, it'd be difficult to transport them. Okay, proteins are the workhorses of living systems. Essentially, anything you can think of as a life process is in all likelihood mediated by a protein. They contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur, and occasionally, depending on their activation state, they might contain phosphorus in the form of a phosphate group. Proteins are polymers. There are about 20 different flavors of amino acids that are used to assemble proteins. A protein is a chain of amino acid that can be any length of any of these 20 building blocks, which can occur in any sequence. So the number of potential molecules that you can build is almost infinite, which is good because they have a lot of jobs to do in the body. When we build a protein, what we do is we hook it together and create a unique covalent bond called a peptide bond, which exists between the carboxylic acid of one amino acid and the amino group of another. And what happens when they come together is they form this unique peptide bond. And we'll see in just a minute how those bonds are created. They contain an amine group and an acid group, and that's what's responsible for their name, amino acid. The amino group is at one end of the monomer, and the acid group is at the other end of the monomer. They can act as either an acid or a base depending on the composition of what we call their R group. Essentially, if we draw a prototypical amino acid, we'll just do this in blue here. This would be what we call the alpha carbon. This would be the acid group, okay, the carboxylic acid group, okay, OH here. And there would be a hydrogen bound to this alpha carbon by a covalent bond. And over here would be an amino group, okay, NH2. Okay. And don't forget the lone pair of electrons here. And then what differentiates the 20 amino acids from each other is their attachment to a variable R group, which we'll indicate in black. And this R group could be a collection of 20 different types of atoms hooked to each other in a variety of different orders. And we'll show you those R groups in just a minute. But basically, if you look at the 20 amino acids, they come in three broad flavors. The nonpolar amino acids, which have R groups that are primarily carbon and hydrogen. The polar amino acids, which contain electronegative atoms as well as carbon and hydrogen, and so they tend to associate more readily with water or other polar amino acid side chains. And then charged amino acids, which at neutral pH will have either a positive or a negative charge, which generally means they have a free amine group or a free carboxylic acid group at the end of this hypothetical R that's attached to the amino acid. And so this dictates essentially how the polypeptide folds up. When you imagine a chain of amino acids, some areas being nonpolar, some areas being polar, and some areas having a positive and others a negative charge, what happens once those amino acids are attached to each other in a long chain is that the R groups will begin to seek each other out, as will components of the polypeptide backbone. And nonpolar groups will seek out nonpolar groups. Polar groups will seek out other polar groups or seek out water. And charged groups will seek out groups of the opposite charge. And what will happen is that the protein will adopt a three-dimensional structure that we call its native conformation, which is the shape in which it's going to perform its job inside the cell. Okay, so these are the different amino acids. All right, so you can see here the charged amino acids, the polar amino acids, and the nonpolar amino acids. The essential amino acids are the ones that our body can't synthesize, so we have to eat these in order to get them, and they include isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, cysteine, and tyrosine, and they're indicated here by the light blue rectangles around the essential amino acids. The other amino acids, we have the metabolic pathways to generate from other building blocks. We can generate them from carbohydrate intermediates in some cases. We can generate them in some cases from breakdown products of other proteins. Basically, we don't have to eat them to get them. But it's this collection of 20 building blocks that allows us to synthesize the wide variety of protein molecules that carry out all the life functions. So let's take a look at how amino acids are synthesized. We have several videos that will touch on different aspects of their structure.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. A hydrogen atom, and 4. A group that differs from one amino acid to another and is represented by the letter R. This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
So the bottom line that we learn from our little mini lesson on proteins is that they have various levels of structure, primary, secondary, tertiary, and quaternary, that give the protein shape, and the shape dictates the function of the protein. And the reason that we're able to build so many different types of molecules using the 20 amino acid building block set is because we can hook them together in any order and in any length we like, and we can even join together, in the case of proteins with quaternary structure, different amino acid chains from completely different, being products of completely different genes, and as a result expand the potential functions of proteins to an even greater degree. Now you might wonder about how it is that this magic sequence of amino acids suddenly dictates a protein's function, and the method by which this occurs is nothing more than evolution and natural selection. Essentially, when a piece of genetic information provides an advantage to a particular organism in that organism's lifestyle or niche, in the sense that it provides it a greater ability to live long enough to reproduce than those organisms around it, then that piece of genetic information is going to be retained, and that protein product is going to be retained, and that information will be passed from that organism down to its progeny, who will inherit the ability to produce this particular protein product that provides that same advantage. And this is a process that is not a simple and short one. It can take millions and millions of years in most cases, and essentially what you're looking at when you look, for instance, at the human body, is the finished product of millions of years of evolution and natural selection to produce a variety of structural proteins, of enzymes, of transport proteins, of defense proteins, of proteins of all ilk that give us the unique advantage to exist in our niche in the environment. And we have now the unique ability, of course, which is in all living things, to pass this information down to our progeny and provide them a greater chance for survival. Okay, so it's not as if you just magically throw random amino acids together and you generate something that's useful to the organism. It's a trial and error type of method. So let's look at some different protein types. Fibrous proteins are strand-like. They tend to be water insoluble primarily due to their size and relatively stable. They generally have tertiary and quaternary structure, so many of them are going to involve more than one amino acid chain. Some involve only a single amino acid chain. They generally have tremendous tensile strength and provide mechanical support, and examples of these include keratin, elastin, and collagen, which is the single most abundant protein in the body. We find it primarily in connective tissue, such as in ligaments or tendons. We also find it in bone, and we also see examples of this in contractile fibers. Examples of contractile fibers would be the proteins actin and myosin. Globular proteins tend to be compact, approximately spherical in shape, but remember that every protein has a native conformation in which it's going to do its job, and that's going to be, in the case of globular proteins, approximately the shape of a particle, but there will be features on the surface of the protein that provide it particular functionality. They're relatively water soluble, primarily due to their small size and also their amino acid content, and they're sensitive to environmental changes. They tend to have tertiary or quaternary structure, and they have specific functional regions. One of the surface features, for instance, that you might find in a globular protein is a crevice. If that protein were, say, an enzyme, that crevice could serve as the catalytic site for that particular enzyme. This would be where the reactants bind, and the enzyme then facilitates the collision of the reactants to produce a product that the cell can use in order to further life processes. Examples of other globular proteins include antibodies, hormones, molecular chaperones, and enzymes. Now, what are each of these? Well, antibodies are the body's equivalent of the magic bullet. A magic bullet would be like a heat-seeking missile. You can aim it in any direction, and it'll always find its target. What the antibody does is it binds to foreign cells, pathogens that enter the body, and tag them for destruction through a variety of methods. They can attract macrophages, which are cells that eat things, and those macrophages can come to the antibody tag pathogen and gobble him up and destroy him. They can immunoprecipitate the pathogen by binding to more than one pathogen at a time and creating such a high molecular weight complex that it comes out of solution, and then wandering macrophages can come by and eat it. They also can stimulate a very important series of chemical reactions in blood plasma called the complement system, which will lead to lysis of the cell that's had the antibody tacked to it. So antibodies are kind of magical things, and we have approximately, well, the potential in anyone's immune system to make close to 1 billion different antibodies in terms of their specificity, their ability to recognize something other than your body's own antigens attack and destroy it. Hormones, of course, are chemical signals. They're produced by special tissues in the body called glands. They're extruded into the extracellular environment, into the bloodstream, where they travel around the body by the pumping action of the heart. And when they arrive at a cell that has a receptor for that particular hormone, that hormone will bind to the receptor. That'll produce a conformational change, which can lead to a response in the target tissue. Hormones that are water soluble tend to have receptors on their target tissues that are on the membrane surface, while hormones that tend to be lipid soluble or hydrophobic tend to have receptors that are inside the cytoplasm of the cell. Molecular chaperones are something that you may not have heard of before. Essentially, what they do is they help very large and complex molecules fold up into their correct conformation. So you could think of them as helpers in the protein's efforts to achieve its native shape. It is an error in a molecular chaperone that actually produces the disease that we know as mad cow disease, or in humans also known as Creutzfeldt-Jacobs disease. It's these misfolded proteins in these diseases that lead to the breakdown of certain cells, among them cells in the nervous system, that leads eventually to degeneration of those structures and death. And of course, enzymes are catalysts for chemical reactions, meaning that they intervene in the process of two reactants coming together to make a product, so that they come together much more efficiently and at a much higher rate of speed, and they produce the product on a much shorter time scale so that the cell can use that product in order to continue its metabolism, continue its life processes. So globular proteins, we couldn't get by without them. Okay, now sometimes what can happen is you can mistreat a protein and cause it to lose its native conformation. This is a process known as denaturation, where the globular proteins unfold and they lose their functional three-dimensional shape. The active sites are destroyed. We can cause this process as a result of a variety of changing conditions around the protein. We can alter the temperature, we can alter the pH, we can alter the salt concentration, we can mechanically mistreat the protein, we can use chemicals that are called chaotropic agents that interfere with the ability of hydrogen bonding to take place successfully. Chaotropic agent literally means to produce chaos, in this case in the protein molecule. And the result is that you've rendered the protein incapable of doing its job. It's usually reversible if we restore normal conditions, but sometimes we can engage in irreversible protein denaturation. And the perfect example of this is if you've ever had a sunny side up egg. When you crack that egg over the hot frying pan, the clear runny fluid is the albumin inside the egg, which is there for nutritive purposes. It's also there to maintain fluid balance. And what happens is that when the albumin hits the hot frying pan, the heat denatures it in an irreversible fashion, converting it from a viscous liquid into a white solid, and there's nothing we can do to get it back into its native conformation after that's happened. And that's partly due to the fact, number one, that the conditions are so severe, but also it's in part due to the size of the protein molecule. Okay, chaperones, again, are globular proteins that allow accurate folding and association of these proteins with other proteins. They prevent incorrect folding, which would produce something not in its native conformation. And they also assist in the movement of proteins and ions across membranes and promote breakdown of damaged or denatured proteins. They're thought to help trigger the immune response. Another type of protein is a stress protein, and what these are a type of chaperone that's made in response to a stressful situation, such as a change in temperature, a change in oxygen concentration, And what they do is they help stabilize cellular functions during stress so that the cell doesn't die. They can delay the aging process by patching damaged proteins and helping them to refold so that they can do their job as well. So you can see that if we have a damaged chaperone, that we could end up now with a sort of a ripple effect through all the proteins in the living cell that could result in rapid degenerative change. And that's exactly what we see in conditions such as mad cow or Creutzfeldt-Jacobs. Remember that enzymes are biological catalysts. They're proteins that increase the speed of chemical reactions and do so by primarily lowering the activation energy, which is the amount of energy needed to really start the chemical reaction. The other thing that they provide is a means of regulating that chemical reaction. Very often when we talk about an enzyme, it'll have not only an active site where the chemical reaction takes place, but it may have one or more regulatory sites which bind to products that are, say, more towards um, the end of a particular metabolic pathway. As those products build up to specific concentrations inside the cell, they can bind to these regulatory sites and they can actually slow um, the catalyst down so that the rate of that chemical reaction slows down so that the cell doesn't waste chemical resources and time making something that it no longer needs. Other regulatory sites might tend to speed up the rate at which the catalyst facilitates that particular chemical reaction. Remember that they lower the activation energy and they increase the speed of the reactions, sometimes on the order of millions of reactions per minute. If we didn't have these important proteins around, the cell simply wouldn't be able to make the products that it needs to live quickly enough to continue as a piece of a living system, and it would cease to exist. So it was the evolution of the catalyst that allowed us to speed up chemical reactions without having to um, change other things that relate to reaction conditions, such as concentration of reactants or temperature. Um, we don't have to do any of that in order for the reaction to proceed at a rate that the living system needs it to, in order for it to be useful to the cell because of the enzyme's presence. Okay, um, this is just a little video that's going to show you a bit about activation energy, so take a listen.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
Okay, some functional enzymes consist of two parts: the apoenzyme, which is the protein portion, and the cofactor, which could be a metal ion or organic molecule, and these are tools that the enzyme can use in order for it to do its job. The coenzymes are very often derivatives of vitamins or the vitamins themselves. The cofactors, metal ions, things like calcium, magnesium, iron, can serve as tools for things like transport proteins or enzyme activity. We get these as a result of our diet. Okay, so this is why you have to consume these trace components in order for metabolism to continue. If the enzyme requires this tool and it doesn't have it available, then the chemical reaction that it catalyzes can't proceed. Enzymes exhibit specificity, meaning that they only act on a particular set of reactants called the substrate, and they're usually named with the name terminating in ASE and the prefix indicating the type of reaction that they catalyze. As an example, hydrolases would be enzymes that catalyze the hydrolysis of large polymers. Oxidases would be enzymes that oxidize organic compounds, very often serving as part of a metabolic pathway that's used to extract energy from that organic compound and transfer it ultimately to something like ATP that the cell can use immediately to do work. And this is just a diagram showing you how the enzyme-substrate interaction works. We have the enzyme, which is the catalyst binding to the substrate on the active site, that does so in an orientation and with sufficient force that these two reactants can collide and create a novel chemical bond, and the result is the release of a product that's useful to the cell. And then look what the catalyst can do. It can go back and recatalyze that exact same reaction until the cell really doesn't need that product anymore, at which point we can either downregulate the activity of that enzyme, or we can quit synthesizing that enzyme and as a result have less of it in the cytoplasm for the reaction to proceed. Now, DNA and RNA are information molecules. DNA is found in two places in the cell. It's found in the nucleus, and it's also found in an organelle called the mitochondria. And it's a large polymer built up of building blocks called nucleotides. RNA is also an information molecule, but it's used primarily to move the information that's contained in the DNA from the nucleus into the cytoplasm of the cell, where it can be read by specialized organelles called ribosomes in order to translate that information into a growing protein. This happens sometimes in the free cytoplasm, and it often happens attached to a very important cellular organelle known as the rough endoplasmic reticulum. DNA and RNA are some of the largest molecules in the body. They contain carbon, oxygen, hydrogen, nitrogen, and phosphorus. They are, of course, polymers. The monomer building block is the nucleotide, which is made up of a nitrogen base, a pentose sugar, and a series of phosphate groups. And it's kind of interesting, if you look at the structure of nucleotides, what you find is that the energy to synthesize the polymer is actually contained in the building blocks. Nucleotides are molecules that are very similar in their structure to the universal energy currency in the body, which is known as ATP. Let's write that up here in red. Okay. And in fact, ATP is an example of a nucleotide. It's a ribonucleotide. The other nucleotides are very similar to ATP. They just vary in terms of the nitrogenous base that's attached to the sugar and to the phosphate group. But it's the triphosphate which is where the energy is in these building blocks. Okay, now the next molecule we want to discuss is DNA, which is one of the two information molecules that we find in living cells. It utilizes building blocks called nucleotides, and a component of each of these nucleotides are what we call nitrogenous bases. And the two nitrogenous bases that are termed purines are adenine and guanine, and the pyrimidines are cytosine and thymine. The base pair rule states that each base is going to pair with its complementary base on the opposite strand of DNA. DNA is an unusual polymer in that it is basically two strands of polynucleotides that run in opposite directions. And when we look at a particular section of a DNA molecule, what we find is that on one strand where we find an adenine, we will always find on the opposite DNA strand a thymine, and where we find a guanine on the opposite strand, we will always find a cytosine. So if we were to sketch this, let's use two different colors for each of our DNA strands. Essentially, what we would have here, one strand would go like so, okay, and then the other strand, which would be the other color, let's make that blue, would go like so. Okay, so these are two strands running in the opposite direction. And then let's say on this strand we have a sequence A, C, A, G. On the opposite strand, we could predict the sequence would be C, T, G, T, because of the base pairing rules. Now the reason that that's important is that when a living system needs to reproduce, when a cell needs to divide from going to be from one cell to being two cells, what happens is that this DNA double helix will unwind. Okay, so let's draw an arrow here indicating this strand unwinding, and another arrow here indicating this strand unwinding. And then let's copy our strand sequences here. So A, C, A, G. And over here, make sure you can see that that's an A. And over here we'd have the opposite strand. And this would be C, T, G, T. And what the cell will do once it unwinds the double helical DNA is it'll come in and it'll make the new strand based off the information that's in the old strand. So if we were to draw the new strands in red, what we'd look at here, we would see C, T, G, T. And of course that strand would run in this direction, always anti-parallel to the original. And then over here we would see A, C, A, G. And lo and behold, what we would have are two brand new double helical molecules, each with the same information content as the original. And so that's the significance of the double-stranded structure of DNA, and that each strand contains information to make the new complementary strand. And that's how information gets passed down faithfully from cell to cell to cell every time we reproduce DNA. A double-stranded helical molecule of DNA is in the cell nucleus, and in the human cell what we have are 46 different DNA molecules that are called chromosomes. You get 23 of those from your dad and 23 of those from your mom. The pentose sugar in the nucleotide is called deoxyribose, and remember that DNA is broken up into information packets that are called genes that contain information for building proteins. It replicates prior to cell division in order to make certain that we pass a faithful copy of the information down to the next cell when it divides. So what happens when a cell divides, as we're going to find out in the next chapter, is that the first thing that occurs is the synthesis of a new copy of genetic material. That genetic material then gets bundled up into chromosomes. Those chromosomes will then be divided evenly between the two new daughter cells, and then the rest of the cellular contents will be divided after the end of mitosis in a process that's called cytokinesis. So mitosis always comes first, cytokinesis most of the time comes second, in probably 99 times out of 100 in human cells, mitosis is followed by cytokinesis. There are a few unusual exceptions that we'll touch on when we actually get to those cell types. And so what you're looking at here is just the structure of DNA. This is a better drawing than what I was showing you. This represents one strand here, okay, and this is its complement. And what we need to understand is that these strands run anti-parallel to each other. Notice that the building blocks, which are shown here, are nucleotides. A nucleotide consists of a phosphate group, a pentose sugar, and this whole thing here would be the nitrogenous base. Okay, this is one of the purines. This is adenine. And over here would be its complementary nucleotide on the opposite strand in the orientation that we'd see it in the double helical molecule. There's the phosphate group, there's the pentose sugar, and there is the nitrogenous base. And this is one of the pyrimidines called thymine. Notice A is pairing with T inside the double helix. Okay, and notice that follows as we go down the DNA molecule, A pairing with T, G pairing with C. Every time we encounter one of these nucleotides, we find its complement on the opposite strand. Okay, and this is how we preserve the integrity of the information. And occasionally what will happen is that a mistake will be made in the duplication of DNA. These mistakes can come from a number of different sources. There are components in the environment that can change DNA structure. They're called mutagens. They can be chemical, or they can come from the exposure to ionizing radiation or UV radiation. Or in some cases, we can alter the information in the DNA as a result of infection by pathogens known as viruses, which can actually insert their own genetic material into our genetic material. The bottom line is that anytime we change DNA structure, we have the potential to change the information that's in the nucleus, and if we do so in a way that's detrimental to the cell, it could result in a variety of different outcomes. It could result in the cell dying in a process known as apoptosis, or it could result in the cell transforming from a normal cell, differentiated cell that's carrying out its task in the human body, into a cancer cell, which would be one that divides without really being concerned about the needs of the organism. And what eventually happens if this cancer cell continues to divide in an uncontrolled fashion is that it will eventually hijack enough oxygen and nutrients away from the organism that the organism won't be able to survive. An unusual property of cancer cells is that they can also leave from their site of origin and spread throughout the body by getting into the lymphatic or the circulatory system, and this is a process known as metastasis. The problem, of course, is that if we don't detect cancer before it has a chance to metastasize, the treatments that we use to deal with cancer are going to be much less effective because of the large number of cells present in the body and the fact that they are occupying now a larger physical space. And so this is why the key to most effective cancer treatment is early detection while the numbers are still low. Another source of mutation is the body's own replication machinery. When we go from making one DNA molecule to making two DNA molecules in what we call the synthesis phase of the cell cycle, occasionally what will happen is that the DNA polymerase will make a mistake and put the wrong base in. Now this is rare, but it does happen. And in the event that this occurs, we have repair enzymes that are inside the cell nucleus ready to fix these mistakes if they happen. If they don't catch these mistakes and the cell does become cancerous, we have another fail-safe, which is the immune system, whose job it is to patrol the body and keep it safe from invasion by pathogens, or to snuff out any cancer cells that crop up on a daily basis. The way that the immune system recognizes these unusual cells is that it checks the cell surface for molecules that are clustered in characteristic arrangements on the cell membrane, which we call antigens. Frequently, cancer cells will have a different set of antigens than normal cells will, and that will mark them for destruction by the immune system. The immune system will move in and, using physical and chemical assaults, attempt to kill that cell before it gets a chance to spread. Generally, we get cancer 2 or 3 times a day, but the reason that most of us are not suffering from widespread metastatic cancer is because we have a fully functioning immune system. One of the things that we see in individuals who have suppressed immunity is increased rates of infection and of cancer. Okay, RNA is the other information molecule in living systems, and it's made up of the bases adenine, guanine, cytosine, and uracil. There is no thymine in RNA. The pentose sugar for the nucleotide building blocks of RNA is ribose as opposed to deoxyribose in DNA. It's still a sugar that has a 5-sided ring, so it's a pentose sugar, but it's got hydroxyl groups attached to most of the carbons as opposed to DNA, which has at one critical position simply a hydrogen atom. So that's one of the ways that we differentiate RNA from DNA, the ribose pentose sugar versus deoxyribose. The use of uracil is also one of the things that differentiates RNA from DNA, and also the fact that it's a single-stranded molecule which does its job mostly outside the nucleus. There's 3 different types of RNA that carry out the DNA orders for protein synthesis. These include messenger RNA, transfer RNA, and ribosomal RNA. So what's the significance of each of these types? Well, let's take messenger RNA first. Okay? Messenger RNA, mRNA, is essentially a copy of the genetic information in the DNA. What happens when a cell needs a particular protein product made is that it will send into the nucleus special proteins that are called transcription factors that will seek out that particular piece of information in the DNA that contains the instructions to make that particular protein. And these transcription factors will open up the double helical molecule so that it's available for an enzyme called RNA polymerase to make an RNA copy off of the coding DNA strand. What will happen is that the RNA polymerase will move from the beginning to the end of the gene. When it gets to a sequence called the terminator, it'll stop synthesizing that particular message, and then that molecule will be processed before it enters the cytoplasm of the cell. One of the ways that it gets processed is that in many genes in eukaryotic cells, we happen to have pieces of DNA that are responsible for coding the protein and pieces of DNA that aren't represented in the protein coding information. So if we were to look, say, at the DNA molecule, the regions that code for the protein are called the exons. The intervening regions are called introns. Okay, and so this would be, for instance, a very simple gene. The boxes here would represent exons, and the lines introns. And so what happens when the RNA polymerase comes in is that he will come in and start to read. So he'll walk from one end of the RNA to the other. And as he does so, he'll make a copy of the DNA that will look exactly like the gene. Okay, so this will be the initial, what we call the nascent RNA molecule. So it will have both exons and introns in it. So it's still happening inside the nucleus. The next thing that will happen is that these intervening pieces of DNA that don't contain protein coding information will be removed. So what we'll do is we'll indicate them as being spliced out using these little red lines here. Okay, so the message will be spliced, and the resulting message will draw here. So it would just be 3 boxes fused to each other. But we're not done yet, because the message still needs a few little modifications. One thing that will happen is that a special cap will be attached to the 5' end of the message, which is made up of a novel nucleotide called 5-methylguanosine. So we'll just write 5-methylcytosine, so 5-MC. And then at the 3' end of the message, what we'll do is we'll tack on a whole bunch of adenine ribonucleotides. This is called polyadenylation. And then what will happen is that this message will be transported out of the nucleus and into the cytoplasm. So cytoplasm out here, CYT, and then all these events here we'll say are happening in the nucleus, NUC. And now this message is going to be available to be used to generate a protein. The instructions in this message will be translated by special organelles called ribosomes into a polypeptide chain that will eventually form a protein that will perform a task that the cell needs it vitally to do. Okay, so there's our 5-methyl cap, and there's our poly-A tail. Okay, and so the next thing that will happen is that this thing will go on to be translated. So translation is something that happens in the cytoplasm, and generating the messenger RNA from the gene is a process that's called transcription, and that only happens in the nucleus. Okay, so transcription here. Okay. Now, how about transfer RNAs? What are they for? Well, transfer RNAs are found in the cytoplasm where they're attached to amino acids, and they're used by the ribosome as it reads the messenger RNA to incorporate amino acids into the growing polypeptide chain. What happens as the ribosome marches down the message is that it will read a sequence of 3 nucleotides hooked together called a codon, and then it will look for the appropriate transfer RNA that is complementary to that codon and incorporate that into the growing polypeptide chain, and then it will move down to the next codon, incorporate the next appropriate transfer RNA and its amino acid, form a peptide bond between the first and second amino acid, release the first transfer RNA minus its amino acid, and then this process will continue until the ribosome gets all the way down to the last codon, which instead of coding for a transfer RNA that contains an amino acid, codes for a transfer RNA that's attached to a water molecule. At that point, the polypeptide chain will be released. It will then adapt its native configuration. The ribosome will then fall off the messenger RNA. The 2 subunits will come apart, and that'll be the end of that particular round of translation. The ribosomal RNA here is a structural component of the ribosome. Okay, so this is a molecule that's associated with ribosomal proteins in a very special area of the nucleus called the nucleolus. And it's the assembled ribosomes that exit through the nuclear pores and then take up their spot either on the endoplasmic reticulum, the rough ER, or free in the cytoplasm where they're called free ribosomes. So those are what the 3 different RNA molecules are designed to do. So let's take a listen to DNA and RNA structure.
DNA and RNA are nucleic acids. polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay. Now, the last molecule we want to talk about is ATP. And the reason ATP is so important is that it's the universal energy currency in living systems. It directly powers chemical reactions in cells that wouldn't otherwise occur. So it's important in metabolic reactions that are what we call anabolic, meaning that we build up large molecules from smaller pieces. It's the energy form that's immediately usable by all body cells, and it has a very characteristic structure. You can see the nitrogenous base, which is adenine, the ribose sugar here, the pentose sugar, and then over here you can see the 3 phosphate groups that are attached to this terminal carbon. This is called the 5' carbon, so 5, 4, and then 3, and then 2 and 1 is the numbering system that we use. These 3 phosphate groups, notice that they have a negative charge, and because negative charges will repel each other, it takes a lot of energy to hold these negative charges together. So there's a lot of chemical potential energy in both of these 2 bonds. And so what happens when this bond is cleaved is that the energy that's released is used to drive reactions that otherwise wouldn't occur spontaneously. So this is critical for anabolic processes to proceed. The best analogy I can draw is the way your car runs. Okay, you know that your car operates on a fuel called gasoline, and gasoline is derived from another fuel that's called crude oil, which comes out of the ground, and it's basically a liquid made up of long chains of hydrocarbons. But if you were to try to put crude oil directly into your gas tank, your engine wouldn't understand that type of fuel and wouldn't be able to run properly. So what we have to do is to process that energy by cracking these long hydrocarbons into smaller molecules, and the it's the 8-carbon hydrocarbon that results from that cracking procedure called octane, which is one of the active components in gasoline, and that's what your carburetor understands. That's what your spark plug can ignite, and that's what your engine is designed to run on in order to convert chemical potential energy into kinetic energy. And the same thing happens in living cells. There's chemical potential energy in fats and in carbohydrates and proteins, but the cell can't use that energy directly to drive work. What it has to do is to break the chemical bonds in those molecules through a process called oxidation, and then transfer that energy ultimately to this molecule, ATP. And it's in this form that we can immediately do work based off of the hydrolysis of these phosphate groups in the ATP molecule. Okay, and granted, there's going to be some energy lost when those conversions take place as heat, but it's still a very efficient way to transfer chemical potential energy from macronutrients to usable energy that the cell needs in order to do work. So what are some of the functions of ATP? Well, it's used for phosphorylation. Sometimes we take the terminal phosphate from ATP and transfer it to other molecules in order to energize them. These molecules can perform cellular work using the energy that comes from the phosphate bond. And the enzymes that do this, that add phosphate groups to proteins to activate them, are called kinases. And then there are other proteins that use ATP in order to do mechanical work, like we can see in this illustration. Here's an example, for instance, of a pump that actively transports a solute from inside the cell to outside the cell against its concentration gradient using the energy derived from ATP. So here we have the membrane protein. There's the pumping action. We release now the inorganic phosphate as we've cleaved that bond that contains the chemical potential energy. We give off the byproducts ADP and inorganic phosphate, and then if we need to continue the process, we'll need to bind another ATP molecule in order to drive this pump so that it can move this solute against its concentration gradient. Another example is the use of ATP to generate kinetic energy. Here we see a relaxed smooth muscle cell, but once we hydrolyze the energy in ATP, the cell is able to contract and shorten, release the byproducts ADP and inorganic phosphate, and the result now is that we've affected movement. And here we can see a process in which we use ATP in order to phosphorylate reactants, providing energy to drive reactions that would normally not be favored energetically. Okay, so we add energy to the left side of the equation so that we can make this high energy product from what were initially low energy reactants A and B, but it's the ATP and its energy in the phosphate bonds that allows this reaction to be pushed over the energy hump so that the cell can generate this useful product. Okay, so hopefully that makes sense. Again, make sure and study this material because it will be covered on the exams and quizzes, and I will see everybody in class. Thank you.